Hello there and welcome to Whispering Universe's channel. Tonight we're diving into the crystalline depths, where sunlight dances through water, exploring the hidden cities beneath the waves. Coral reefs stand as underwater cathedrals, built grain by grain over millennia by tiny architects working in perfect harmony. These living monuments rise from the seafloor like dreams made manifest, their spires and buttresses crafted not from stone and mortar, but from the accumulated life force of countless generations. These living rainforests of the sea teem with secrets we are only beginning to understand. They harbour ancient wisdom in their calcium carbonate walls, shelter countless creatures in their branching embrace, and pulse with rhythms as old as the ocean itself. Each coral colony represents a testament to the power of collective action, where millions of individual polyps work together to create structures that dwarf the greatest human achievements. From the tiniest polyp to the vast networks that span continents, coral reefs whisper stories of cooperation, survival, and breathtaking beauty. They speak in languages of colour and chemistry, in patterns of growth that follow mathematical principles we're only now beginning to decipher. Most of the time, we see only their surface beauty. We admire their colours, marvel at their fish, and appreciate their protection of our shorelines. We photograph their brilliant inhabitants and dive among their formations, experiencing just the thinnest slice of their complex existence. But coral reefs are far more than living ornaments. They are complex cities with sophisticated infrastructure, chemical laboratories where nature experiments with new compounds, weather predictors that sense atmospheric changes days in advance, and time capsules that preserve the history of our oceans in their very structure. They communicate across vast distances through chemical signals that drift on ocean currents, wage wars in slow motion using weapons more sophisticated than anything in human arsenals, and create their own light shows in the depths using proteins that have revolutionized medical research. Science is revealing their world to be far more intricate than we ever imagined. Each discovery opens new chapters in the story of these underwater metropolises, where cooperation and competition interweave to create some of Earth's most spectacular ecosystems. Every research expedition returns with findings that challenge our understanding of what these organisms are capable of. The more we learn about coral reefs, the more we realize how much we still don't know about their hidden complexities. They are living libraries of evolutionary innovation, archives of climate history, and laboratories where the fundamental processes of life play out in ways that continue to surprise and inspire scientists around the world. Beneath the waves, millions of tiny architects work without rest, each one no larger than a rice grain, yet together they construct monuments that dwarf human cities and can be seen from space. These coral polyps, simple animals with no brains or hearts, possess an intelligence that emerges from their collective actions. They are master chemists, extracting minerals from seawater with precision that industrial processes struggle to match, and transforming them into living stone that will endure for millennia. They are urban planners, creating complex three-dimensional structures that maximize space utilization and provide homes for thousands of species. They are farmers, cultivating algae in their tissues with care that rivals any agricultural system, and harvesting sunlight to feed entire ecosystems through an ancient partnership that transformed the oceans. The coral reef cities never sleep. As daylight fades and the last rays of sun filter through the water, painting the reef in deep blues and purples, a transformation begins. Night shift workers emerge from their hiding places, octopuses unfurl their arms from rocky crevices like liquid sculptures, eels glide through coral corridors with serpentine grace, and countless small creatures begin their nocturnal journeys through the maze of calcium carbonate. The coral polyps themselves extend their feeding tentacles, transforming the reef from a landscape of stone into a living meadow of swaying arms that dance in the current, each one a tiny hunter waiting for plankton to drift within reach. In the darkness, bioluminescent displays flicker like underwater fireworks, creating a light show that rivals any terrestrial display. Plankton spark blue-green when disturbed, creating trails of light behind swimming fish like comet tails in a liquid sky. Some corals glow with their own ethereal light, revealing patterns invisible during the day, secret messages written in fluorescence that speak to creatures whose eyes can see beyond human perception. The reef at night is a different world entirely, filled with sounds and lights and movements that most humans never witness, a parallel universe that exists in the same space but operates by different rules. But even in daylight, the reef holds countless mysteries. Every surface teems with life, invisible to casual observation. Entire civilizations of organisms living at scales we can barely imagine. Bacteria form complex communities that process nutrients and fight diseases with sophisticated chemical warfare. Microscopic organisms drift through the water column like living snow, forming the base of food webs that support everything from tiny fish to massive whale sharks. The chemistry of reef waters is precisely balanced through the metabolic symphony of millions of organisms, maintained by the metabolic activities of millions of organisms working in concert, each one playing its part in a biochemical orchestra that never stops performing. As always, if you enjoy drifting off to science and wonder, be sure to like and subscribe or share a thought down below. But for now, let the surface tensions fade away. Allow yourself to sink into the gentle current of discovery. Breathe slowly like the ocean's rhythm, and let your mind drift among the whispered wonders of coral reefs. Release the weight of the day and let it dissolve into the vast blue. Tonight, we explore not just an ecosystem, but a way of being, one where cooperation creates strength, where beauty emerges from function, and where the smallest actions accumulate into monuments of life. Let's begin. Though they sway like underwater flowers and remain rooted in place, corals are animals belonging to the same group as jellyfish and sea anemones. This classification places them within the phylum Cnidaria, an ancient lineage of animals that first appeared in Earth's oceans over 500 million years ago. Each coral head is actually a colony of thousands of tiny creatures called polyps, no larger than a pencil eraser, sometimes smaller than a pinhead. These simple animals lack brains, hearts, or blood, yet together they construct some of Earth's most magnificent structures through a form of collective intelligence that emerges from their coordinated actions. Each polyp is essentially a small sac with a mouth surrounded by tentacles, a body plan so simple yet so successful that it has persisted virtually unchanged for hundreds of millions of years. They capture plankton and small fish with stinging cells called nematocysts, just like their jellyfish cousins. These microscopic harpoons are marvels of biological engineering, capable of firing with explosive force in microseconds, faster than a bullet leaving a gun. At night, when the reef comes alive with feeding, millions of these tiny tentacles emerge like a meadow of sea anemones, creating a living carpet that filters the ocean's bounty. The tentacles wave in synchronized patterns, responding to chemical cues and water movements, creating underwater choreography that is both functional and beautiful. The confusion about their nature is understandable and has persisted through centuries of human observation. Corals don't move like typical animals, and many host photosynthetic algae that give them plant-like qualities. Early naturalists classified them as plants, or even as minerals, unable to reconcile their stone-like appearance with their animal nature. But beneath their seemingly passive exterior lives a complex animal society, each polyp connected to its neighbors through living tissue called choanosarc, sharing nutrients and information across the colony like a biological internet. This connecting tissue allows the colony to function as a superorganism, with individual polyps specialized for different tasks, some focused on feeding, others on defense, and still others on reproduction. This animal nature explains much about their behavior that would be inexplicable if they were plants. They can sense changes in light, temperature, and water chemistry with remarkable precision. Their sensory capabilities allow them to detect approaching predators, the chemical signatures of prey, and even the health status of neighboring coral colonies. They respond to threats by closing their polyps or releasing chemical signals that can trigger defensive responses across entire reef sections. Some species even fight their neighbors for territory, extending special sweeper tentacles armed with concentrated batteries of stinging cells in slow-motion battles that can last for weeks or months, reshaping the reef's landscape through chemical warfare. To understand coral reefs is to appreciate this fundamental truth. You are observing not a garden, but a city of millions of small animals working together, each one a master architect, contributing to structures that can be seen from space. The polyps work continuously, secreting calcium carbonate from dissolved minerals in seawater with a precision that human technology struggles to replicate, building their limestone homes with the dedication of master craftsmen. Each polyp maintains its own small territory within the colony while contributing to the collective structure, creating a balance between individual needs and colonial success. Their collective effort creates the foundation for entire ecosystems, supporting more biodiversity per square meter than almost any other habitat on Earth. The individual polyps themselves are marvels of biological engineering, demonstrating that complexity doesn't always require complicated anatomy. Despite lacking a centralized nervous system, they coordinate their activities through chemical signals and direct cell-to-cell communication transmitted through their connecting tissues. When one polyp detects food, it can signal its neighbors to extend their tentacles as well, creating coordinated feeding waves across the colony surface that maximize capture efficiency. This coordination extends to defense as well. When one part of a colony is attacked, chemical alarm signals spread rapidly through the choanosac, triggering protective responses throughout the entire colony. Each polyp operates its own miniature life support system, taking in water through its mouth to circulate nutrients and oxygen throughout its simple body cavity called the colentron. They can expand and contract their tissues to regulate water flow and optimize feeding conditions, adjusting their body volume by up to 50% to accommodate changing environmental conditions. Some species can even move their entire bodies slightly to better position themselves for feeding or to avoid debris and sediment, demonstrating a level of behavioral sophistication unexpected in such simple organisms. The polyps' mouths serve multiple functions beyond feeding, acting as multifunctional portals to the outside world. They release waste products, expel reproductive material during spawning events, and can even eject harmful substances or damaged tissue when necessary through a process called mesenterial extrusion. This multifunctional opening represents the polyp's primary interface with the external world, managed by rings of muscles that can close tightly to protect the polyp, or open wide to engulf surprisingly large prey. Within the simple body of each polyp lies a sophisticated biochemical factory, more complex than any human industrial facility operating at that scale. Special cells called cnidocytes contain the stinging structures that corals use to capture prey and defend themselves. These microscopic harpoons can be fired with explosive force, injecting toxins into target organisms with precision that would be impressive in any predator. The precision and speed of these cellular weapons rival the most advanced human-made projectiles, demonstrating that evolution has created biological machinery of astounding sophistication, even in these simple animals. The digestive system of coral polyps is remarkably efficient, capable of breaking down a wide variety of organic matter using powerful enzymes. They can digest everything from microscopic bacteria to small fish, extracting maximum nutrition from whatever food sources are available in the nutrient-poor tropical waters. This metabolic flexibility allows corals to survive in the nutrient-poor waters of tropical seas, where other organisms would starve, making them masters of efficiency in one of Earth's most challenging marine environments. Every coral reef is essentially a graveyard turned into a thriving metropolis, a testament to how life transforms death into the foundation for new existence. The beautiful formations we see are constructed from the calcium carbonate skeletons of countless coral polyps that lived and died over thousands of years, each one adding its small contribution to an eternal monument. Each generation builds upon the bones of its ancestors, creating layer upon layer of limestone architecture that records the passage of time in stone. This process transforms mortality into permanence, creating structures that outlast civilizations and witness the slow dance of geological change. When a coral polyp dies, its soft tissue decomposes, returning its organic components to the reef ecosystem, but its hard skeleton remains as an eternal testament to its brief life. New polyps settle on these foundations, secreting their own limestone cups and adding to the structure with mathematical precision. The young polyps don't simply rest on their ancestors' remains. They actively incorporate them into their growing colony, creating seamless transitions between the living and the dead. Over time, this process creates massive formations that can stretch for kilometers and rise hundreds of meters from the seafloor. The Great Barrier Reef, for instance, contains the skeletons of corals that lived during the last ice age. Their calcium carbonate remains now supporting entirely new ecosystems built upon ecosystems, a vertical history written in limestone. This constant construction makes coral reefs among the most enduring biological structures on Earth, rivaling the longevity of the oldest forests and exceeding them in structural permanence. While forests can be destroyed by fire and replanted within decades, coral reefs represent millennia of accumulated growth that cannot be quickly replaced. Each storm that breaks coral branches, each predator that consumes coral polyps, adds to the rubble foundation that future generations will colonize and build upon. A coral head that appears to be a single organism may contain the skeletons of hundreds of generations, each one contributing to the architectural marvel we see today, a living cathedral built on the foundations of its own history. The process is remarkably efficient, representing one of nature's most elegant recycling systems. Corals extract calcium and carbonate ions directly from seawater, where these minerals exist in relatively low concentrations, concentrating these dissolved minerals into solid limestone with an efficiency that human technology cannot match. This biochemical process removes carbon from the ocean, making corals important players in global carbon cycling and climate regulation. Each tiny polyp is essentially a living limestone factory, working day and night to build and maintain the reef, adding approximately 0.3 to 2 centimeters of new skeleton per year, depending on the species and environmental conditions. The accumulated work of billions of these tiny factories creates structures that alter ocean currents, weather patterns, and the distribution of marine life across entire ocean basins. Walking through a limestone cave or examining a piece of coral rock on a beach, you are holding the compressed history of an underwater civilization, the accumulated legacy of millions of tiny animals who spent their brief lives adding to something far greater than themselves. Each layer tells a story. Rapid growth during favorable periods appears as widely spaced growth bands, while stress periods leave tight, compressed layers that speak of challenging times. The ancient layers tell stories of past climate conditions, recording changes in ocean temperature and chemistry with the precision of geological chronometers. Scientists can read these stone libraries to understand how ocean conditions have changed over centuries, detecting ancient El Niño events, volcanic winters, and periods of unusual warmth or cold, making coral reefs among our most valuable archives of Earth's environmental history. The calcification process itself is a wonder of biochemistry that scientists are still working to fully understand. Corals must extract calcium ions from seawater, where they exist in relatively low concentrations, actively pumping them across cell membranes against concentration gradients, then combine them with carbonate ions to form calcium carbonate crystals in precisely controlled conditions. This process requires significant energy and is sensitive to ocean chemistry changes, particularly ocean acidification, which reduces the availability of carbonate ions and makes it harder for corals to build their skeletons. The polyps create specialized compartments where they can control pH and ion concentrations with precision that would be impressive in any laboratory, let alone in a tiny animal without a brain. Different coral species create skeletons with distinct architectures, each one optimized for its particular ecological niche. Brain corals form rounded, boulder-like structures with intricate surface patterns that resemble the folds of a human brain, maximizing surface area while maintaining structural strength. Staghorn corals create branching formations that provide complex three-dimensional habitat, but sacrifice durability for rapid growth. Table corals grow flat, plate-like structures that maximize surface area for their photosynthetic partners, while creating shelter underneath for fish and invertebrates. Each architectural style represents millions of years of evolutionary refinement, creating forms that balance growth rate, structural integrity, and ecological function. The strength and durability of coral skeletons rival those of human-made concrete structures and in many ways exceed them. The crystalline structure of calcium carbonate, organized by biological processes with molecular precision, creates materials that can withstand enormous pressure and stress while remaining surprisingly lightweight. Some coral skeletons are so durable that they persist as recognizable structures for millions of years in the fossil record, allowing us to study reef ecosystems from eras when the continents were in different positions and the climate was vastly different from today. The architecture of coral skeletons follows mathematical principles that optimize strength while minimizing material use, demonstrating nature's engineering genius. The intricate patterns of ridges, valleys, and chambers in coral skeletons distribute stress efficiently and provide maximum surface area for the living tissue above. These natural engineering solutions have inspired human architects and engineers seeking to create more efficient building designs, leading to innovations in everything from bridge construction to spacecraft design. The study of coral skeleton architecture has revealed that these simple animals have solved complex engineering problems that human technology is only beginning to appreciate. One of nature's most successful partnerships plays out in the tissues of every healthy coral, a collaboration so intimate that the partners function almost as a single organism. Microscopic algae, called zooxanthellae, live inside coral polyps, creating a relationship that has persisted for millions of years and fundamentally transformed Earth's oceans. These tiny plants, no bigger than bacteria and often smaller than red blood cells, are what give most corals their vibrant colors and provide up to 90% of their energy needs. Without them, the spectacular coral reef ecosystems that harbor a quarter of all marine species could not exist in the nutrient-poor tropical waters where they thrive. The arrangement is remarkably sophisticated, representing one of evolution's most elegant solutions to the challenge of surviving in nutrient deserts. The coral provides the algae with a protected home inside specialized cells called zooxanthellae, safe from predators and environmental stresses, and nutrients like nitrogen and phosphorus from its waste essentially fertilizing its internal gardens with its own metabolic byproducts. The coral's tissues also concentrate carbon dioxide for the algae, enhancing their photosynthetic efficiency beyond what they could achieve in open water. In return, the algae photosynthesize, converting sunlight into sugar and oxygen that feeds their coral host, producing as much as 95% of the coral's daily energy requirements during peak sunlight hours. This partnership allows corals to thrive in nutrient-poor tropical waters, where other organisms struggle to find enough food, turning oceanic deserts into underwater rainforests. Different coral species host different types of zooxanthellae, and this diversity is far greater than scientists initially realized. Recent genetic studies have revealed that what was once thought to be a single species of algae is actually a diverse group of organisms, each with unique characteristics and environmental tolerances. Some corals can even switch between algae types depending on environmental conditions, a flexibility that may be crucial for surviving climate change. When waters become too warm, corals often expel their algae partners, leading to coral bleaching, a dramatic stress response that can be fatal if prolonged. This isn't always fatal. Corals can survive for weeks or months on their own reserves and may take up new algae when conditions improve, sometimes acquiring different, more heat-tolerant strains that better suit the new environmental conditions. The partnership is so intimate that neither organism can reach its full potential alone, representing a form of co-evolution so complete that the partners have become interdependent. Corals without zooxanthellae grow much more slowly, sometimes at only 10–20% of their normal rate, and struggle to compete for space on crowded reefs. They must rely entirely on capturing prey for nutrition, a challenging task in the nutrient-poor waters where most coral reefs develop. The algae, meanwhile, have evolved to live only inside coral tissues, losing the ability to survive independently in the open ocean, where they would be consumed by filter feeders or unable to access the nutrients they need. Many strains of zooxanthellae have lost genes necessary for free-living existence, becoming permanent residents of their coral hosts. This ancient alliance explains why coral reefs are found primarily in shallow, sunlit waters with remarkable clarity. Like underwater forests, they depend on photosynthesis to fuel their growth, requiring clear water that allows sunlight to penetrate to depths where corals grow. The partnership transforms barren tropical seas into some of the most productive ecosystems on Earth through the power of cooperation. The algae work continuously during daylight hours, converting carbon dioxide and water into sugars using the energy of sunlight with an efficiency that can exceed 90%, while the coral provides protection and concentrates nutrients for optimal algae growth. This biological solar power plant operates with an efficiency that human technology struggles to match, converting more of the sun's energy into usable biomass than most terrestrial ecosystems. The relationship between corals and zooxanthellae represents one of evolution's greatest success stories, enabling both partners to colonize environments that neither could survive in alone. The coral gains access to the abundant energy of sunlight without having to develop its own photosynthetic machinery, while the algae receive protection from grazers and access to essential nutrients that are scarce in tropical waters. This partnership has persisted through mass extinctions, ice ages, and periods of dramatic climate change, demonstrating remarkable resilience over geological timescales. Research has revealed that there are actually many different species of zooxanthellae, belonging to multiple genera within the family Symbiodiniaceae, each with specific temperature tolerances and ecological preferences. Some corals can host multiple algae species simultaneously, creating diverse internal ecosystems within their tissues that may function like insurance policies against environmental change. This diversity may provide insurance against environmental change, as corals can potentially shift their algae communities to better match changing conditions, a process called symbiont shuffling that may be crucial for reef survival in warming oceans. The density of zooxanthellae within coral tissues is carefully regulated through sophisticated feedback mechanisms. Too few algae and the coral doesn't receive enough energy from photosynthesis, potentially starving in the nutrient-poor tropical waters. Too many algae and they begin competing with each other for nutrients, potentially becoming parasitic rather than beneficial, extracting more resources than they provide. Corals maintain optimal algae densities through complex biochemical signals that control algae reproduction and survival, including the selective digestion of excess algae and the regulation of nutrient supply to control algae growth rates. The exchange of nutrients between corals and their algae involves sophisticated molecular transport systems that scientists are still working to understand fully. Sugars produced by algae photosynthesis are actively transported to coral cells through specialized carrier proteins, while nitrogen and phosphorus compounds flow in the opposite direction through different transport pathways. This metabolic integration is so complete that coral and algae function almost as a single organism, sharing not just nutrients, but also cellular signals that coordinate their activities. The partnership involves the exchange of hundreds of different compounds, creating a metabolic network of stunning complexity. Despite covering less than 1% of the ocean floor, coral reefs support approximately 25% of all marine species, a concentration of life that defies conventional ecological expectations. In terms of biodiversity per unit area, they exceed even tropical rainforests, the terrestrial champions of biological diversity that have long captured our imagination. A single coral reef system can host thousands of species of fish, invertebrates, and other marine life in an area no larger than a city block, creating living communities so complex that scientists are still discovering new interactions and relationships after decades of study. This extraordinary density of life makes coral reefs the most biologically productive ecosystems in the marine realm, and among the most valuable in terms of ecological services. This incredible diversity stems from the reef's three-dimensional structure and the countless ecological niches it creates through its complex architecture. Every surface, crevice, and overhang provides habitat for different creatures, from microscopic bacteria to massive groupers. The reef structure creates gradients of light, water flow, and shelter that allow species with vastly different requirements to coexist in close proximity. Fish that feed on plankton in the water column live alongside species that graze algae from coral surfaces, hunt in sandy channels, or hide in coral branches waiting to ambush prey. Each level of the reef, from the shallow reef crest exposed to pounding waves to the deeper slopes where light barely penetrates, supports distinct communities adapted to different light levels, wave conditions, and predation pressures. This vertical zonation multiplies the available habitat within a given area, allowing reefs to support far more species than a flat substrate of similar size. The reef's productivity sustains this diversity through an intricate web of energy transfer and nutrient cycling. The partnership between corals and their algae creates so much food that complex food webs can flourish even in waters that are otherwise biological deserts. Primary production from zooxanthellae photosynthesis cascades through the ecosystem, supporting herbivores, which feed carnivores, which feed apex predators, creating trophic pyramids of stunning complexity. Tiny creatures feed on coral mucus, which is constantly produced and shed into the water column. Larger fish hunt the small ones in an endless dance of predator and prey, and cleaning stations develop where fish wait in line to have parasites removed by specialized cleaner species that have evolved specifically for this role. Decomposers break down organic matter and recycle nutrients with remarkable efficiency, ensuring that nothing is wasted in these nutrient-limited systems. Many reef species are highly specialized, found nowhere else on Earth, having evolved unique adaptations to exploit specific reef microhabitats. Some gobies live only on specific coral species, their entire evolutionary history tied to a single type of coral host. Certain shrimp form exclusive partnerships with particular sea anemones, their bodies perfectly adapted to live among the stinging tentacles that would kill other creatures. This specialization creates a web of interdependence that makes reefs both resilient and vulnerable, but resilient because of their complexity and redundancy, vulnerable because the loss of any key species can cascade through the system, potentially triggering ecosystem-wide changes. Each species represents a unique solution to the challenge of surviving on the reef, and many have evolved relationships so specialized that they cannot exist without their specific partners. Scientists estimate that we have discovered only a fraction of reef species, perhaps as little as 10–20% of the total diversity. Each expedition to remote reefs reveals new fish, crustaceans, and other creatures, suggesting that coral reefs harbor more biodiversity than we currently realize. The cryptic nature of many reef inhabitants means that a coral head the size of a washing machine may contain hundreds of species, most of them unknown to science, living in microscopic spaces within the coral skeleton or hiding in crevices too small for human observation. This hidden biodiversity includes bacteria, archaea, viruses, and microscopic organisms that play crucial roles in reef function but remain largely unstudied. Recent advances in genetic sequencing have revealed that the microbial diversity alone on coral reefs may exceed that of all visible organisms combined. The vertical zonation of coral reefs creates distinct habitat layers, each supporting different communities adapted to specific conditions. The reef crest, constantly pounded by waves, hosts robust, wave-resistant species that can withstand the constant mechanical stress. The protected back reef areas support delicate branching corals and their associated fish communities, creating nursery areas for many species. The reef slope provides gradients of light and water movement that create numerous microhabitats, each with its own unique assemblage of species. This three-dimensional complexity is what allows so many species to coexist in such a small area, with each species occupying its own ecological niche. Within each zone, the complex architecture of coral growth creates countless microenvironments that further multiply the available habitat. A single large coral head may provide habitat for boring organisms that excavate tunnels through the skeleton, cryptic species that hide in crevices during the day and emerge at night, and epiphytic organisms that grow on coral surfaces without harming their host. This architectural complexity is what allows reefs to pack so much biodiversity into relatively small areas, creating ecological communities of unparalleled richness. The surfaces of corals themselves are ecosystems in miniature, hosting communities of bacteria, algae, and microscopic invertebrates that form complex food webs invisible to the naked eye. The diversity of coral reefs extends beyond visible organisms to include vast communities of microbes that play essential roles in reef function. Bacterial communities associated with corals help with nutrient cycling, disease resistance, and chemical communication between reef organisms. The coral holobiont, the coral animal, plus all its associated microorganisms, functions as a complex ecosystem in miniature, with different microbial species performing specialized functions that contribute to coral health and survival. These microbial partners may produce antibiotics that protect corals from disease, fix nitrogen from seawater to provide essential nutrients, or help corals adapt to changing environmental conditions through rapid evolution. Seasonal changes in reef biodiversity can be dramatic, with different species dominating at different times of year based on reproductive cycles, food availability, and environmental conditions. Spawning aggregations bring together enormous numbers of fish during reproductive seasons, temporarily increasing local biodiversity to extraordinary levels. Larval settlement events introduce new cohorts of young organisms that dramatically alter community composition, creating pulses of recruitment that maintain population diversity. Migration patterns bring seasonal visitors to reefs, from massive whale sharks that arrive to feed on spawning aggregations to seabirds that nest on nearby islands and feed in reef waters. These temporal dynamics add another dimension to reef biodiversity, creating communities that are constantly changing in composition while maintaining their overall structure and function. While individual coral polyps may live only a few years, coral colonies can achieve remarkable longevity through continuous growth and regeneration, creating organisms that persist across millennia. Through a combination of clonal reproduction and cellular regeneration, some coral colonies have been growing continuously since before the rise of human civilization. The oldest known coral colonies are massive brain corals and boulder corals that have been growing for millennia, their ancient cores bearing witness to centuries of ocean history. These living monuments have survived ice ages, volcanic eruptions, and countless storms, adapting to changes that would destroy most other organisms. Black corals, despite their name referring to their dark skeletons rather than their living tissue, often live the longest of all coral species. Some deep-water black coral colonies in Hawaii are estimated to be over 4,000 years old, making them among the oldest living organisms on Earth, older than the giant sequoias of California and rivaling the ancient bristlecone pines. These ancient animals were already mature when the pyramids were built, when bronze was first being worked by human hands, and they continue growing today in the dark depths where few other organisms can survive. Their growth is incredibly slow, sometimes adding less than a millimeter of skeleton per year, but their persistence transforms them into living chronicles of ocean history. The longevity of corals comes from their unique biology and their remarkable ability to regenerate damaged tissue. Unlike most animals, corals can theoretically live forever through asexual reproduction, constantly replacing old polyps with new ones that are genetically identical. When a polyp divides, it creates genetically identical clones, allowing the colony to maintain genetic continuity across vast time spans that encompass the rise and fall of civilizations. Damage to part of the colony doesn't kill the whole organism. Healthy sections continue growing and may eventually recolonize damaged areas, demonstrating a form of biological immortality that few other organisms possess. This modular construction means that while individual polyps age and die, the colony as a whole can persist indefinitely, constantly renewing itself through cellular regeneration and growth. These ancient corals serve as living libraries, their skeletons recording changes in ocean temperature, chemistry, and climate over centuries with the precision of scientific instruments. By analyzing the chemical composition of coral skeletons, scientists can reconstruct past environmental conditions, studying ancient El Niño events, volcanic eruptions, and other climate phenomena from long before human records began. Each year of growth adds a new layer to this environmental archive, preserving information about water temperature, salinity, nutrient levels, and even the frequency of storms. The isotopic signatures in their skeletons reveal ocean temperatures from centuries past, with precision that rivals modern instruments, while trace elements record pollution levels, river runoff, and other environmental changes. To encounter one of these ancient colonies is to meet a being that has outlasted empires and witness the rise of human civilization, growing grain by grain in the eternal rhythm of the sea. They were young when Rome ruled the Mediterranean, mature when Vikings sailed the North Atlantic, and ancient when Columbus crossed the ocean. Their growth rings tell stories of drought and plenty, of volcanic winters that darkened skies around the globe and warm periods that melted polar ice, of ocean changes that occurred when humans were still learning to build their first cities. The chemical signatures locked in their skeletons reveal not just temperature, but also the composition of ancient atmospheres, the intensity of past hurricanes, and the effects of long-ago volcanic eruptions that changed global climate. The study of coral longevity has revealed fascinating insights into biological aging and the mechanisms that allow some organisms to escape the senescence that affects most animals. Unlike most animals, which show clear signs of senescence as they age, declining reproduction, slower growth, the increased susceptibility to disease, ancient coral colonies often continue growing at steady rates and maintaining healthy tissue even after centuries or millennia of life. This apparent resistance to aging makes corals valuable subjects for research into longevity and cellular maintenance, potentially offering insights that could benefit human medicine. The cellular mechanisms that allow corals to maintain themselves across such vast time spans involve sophisticated DNA repair systems, antioxidant defenses, and the ability to replace damaged cells with healthy ones. Some of the most ancient corals have survived multiple major disturbances, including volcanic eruptions that acidified oceans, severe storms that shattered reefs, and dramatic climate changes that caused sea levels to rise and fall by hundreds of meters. Their persistence through these challenges demonstrates the remarkable resilience that can be achieved through longevity and slow, steady growth. These survivors have witnessed the extinction of countless other species, the evolution of new forms of life, and the transformation of ocean ecosystems over geological timescales. Their survival strategies include the ability to enter dormant states during unfavorable conditions, to regenerate from small surviving fragments, and to adapt their growth patterns to changing environmental conditions. Ancient coral colonies often serve as foundations for entire reef ecosystems, providing stable substrate that allows other organisms to establish long-term communities. Their massive skeletons create the three-dimensional structure that supports thousands of other species, from tiny bacteria to large fish. The three-dimensional structure, created by millennia of coral growth, creates complex habitat that supports biodiversity over geological timescales, with some reef communities maintaining similar species compositions for thousands of years. These ancient colonies are like old-growth forests of the sea, creating stable ecosystems that support far more species than younger reef areas. The environmental conditions that allow corals to achieve extreme longevity are specific and becoming increasingly rare in our rapidly changing oceans. Stable temperatures within a narrow range, clean water free from pollution and excessive nutrients, and protection from physical disturbance are all necessary for corals to reach their maximum lifespans. Climate change and human impacts are threatening these conditions, potentially ending the era of ancient coral colonies that has persisted since before human history began. The loss of these ancient organisms would represent not just an ecological tragedy, but the destruction of irreplaceable environmental archives that can never be reconstructed. Once or twice a year, usually triggered by moon phases and water temperature, many coral species participate in one of nature's most spectacular reproductive events. A synchronized ballet of reproduction that transforms entire reef systems. On a single night, millions of coral polyps simultaneously release eggs and sperm into the water, creating underwater blizzards of reproduction that can be seen from space as milky clouds drifting across the ocean surface. This mass spawning represents one of the most precisely coordinated biological events on Earth, with colonies separated by kilometers, releasing their gametes within minutes of each other, as if responding to an ancient signal that resonates through the reef. This mass spawning is precisely timed through a complex interaction of environmental cues that corals have evolved to detect with remarkable sensitivity. Different coral species release their gametes hours apart, reducing competition and hybridization while maximizing the chances of successful fertilization within species. Some spawn just after sunset, when the water is calm and predators are changing shifts. Others in the early hours of morning, when tidal currents are optimal for dispersal, each following an internal clock synchronized to lunar cycles and seasonal changes. The coordination is so precise that marine biologists can predict spawning nights weeks in advance, planning research expeditions around these brief windows of reproductive activity. Water temperature must be within a specific range, usually after it has reached a seasonal maximum and begun to cool slightly, while the moon phase provides the final trigger that initiates the spawning cascade. The strategy behind mass spawning is brilliant in its simplicity and effectiveness. By overwhelming the water column with reproductive material all at once, corals ensure that many eggs and sperm will find each other before being eaten by the countless filter-feeding creatures that normally patrol the reef. The sheer volume of genetic material released creates better odds for successful fertilization through a phenomenon called predator satiation. There is simply too much food available for predators to consume it all. This reproductive strategy, known as broadcast spawning, allows corals to achieve fertilization rates that would be impossible if they released gametes gradually over time. The synchronized timing also ensures that eggs and sperm from the same species are in the water simultaneously, increasing the likelihood of successful fertilization despite the vast dilution of the ocean. The larvae that result from this spawning drift in the ocean for days or weeks before settling on hard surfaces to begin new colonies, embarking on one of nature's most perilous journeys. This larval stage allows corals to colonize distant reefs, maintaining genetic connections across vast ocean basins and preventing the isolation that could lead to inbreeding. Some coral larvae can survive in the plankton for months, crossing entire oceans to establish new populations on remote reefs thousands of kilometers from their parents. During this time, they must avoid predation, find enough food to sustain themselves, and navigate ocean currents to find suitable settlement sites, a challenge that only a tiny fraction will survive. Witnessing a coral spawning is like watching the reef's dreams made manifest. Millions of potential futures drifting upward through moonlit waters, each one carrying the genetic wisdom of ancient colonies into an uncertain future. The water becomes thick with reproductive material, creating a soup of life so dense that visibility can drop to mere centimeters. This creates feeding frenzies among fish, invertebrates, and plankton that transform the entire reef ecosystem for days following the spawning event. Whale sharks, manta rays, and other large filter feeders often arrive at reefs during spawning season, timing their migrations to coincide with this bonanza of nutrition. The timing of these events influences the reproductive cycles of many other reef organisms, creating cascade effects that ripple through marine food webs for weeks or months afterward. The preparation for spawning begins weeks or even months before the actual event, requiring substantial energy investment from the coral colonies. Coral polyps must allocate significant energy to producing eggs and sperm, often reducing their growth rates during the reproductive season, as resources are diverted to gamete production. The eggs are particularly energy-rich, packed with lipids and proteins to nourish the developing larvae during their planktonic journey when they cannot feed themselves. Some species invest so heavily in reproduction that they may not spawn every year, instead accumulating energy over multiple years for a single massive reproductive effort. The development of reproductive cells within coral polyps follows precise developmental schedules, with eggs and sperm maturing in synchrony across entire colonies and even between colonies of the same species. The environmental cues that trigger spawning are complex and species-specific, involving multiple sensory systems that scientists are still working to understand fully. Most corals respond to the combination of rising water temperatures that indicate the proper season, lunar cycles that may help with tidal timing, and seasonal photoperiod changes that mark the progression of the year. Some species may also detect chemical cues from other spawning corals, creating a cascade effect across the reef. The precise timing ensures that spawning occurs during optimal conditions for larval survival and settlement, usually during periods of calm weather when water mixing is minimal and larvae won't be swept away from suitable habitat. Different coral species use different spawning strategies that reflect their evolutionary history and ecological requirements. Some release bundles containing both eggs and sperm that break apart at the surface, a strategy that may increase fertilization rates, while others release eggs and sperm separately in clouds that must find each other in the water column. Some species are hermaphroditic, producing both types of gametes from the same colony, while others have separate male and female colonies that must coordinate their spawning for successful reproduction. A few species have evolved internal fertilization, brooding their larvae within the polyp before releasing fully developed planulae, ready to settle immediately. Each strategy represents a different solution to the challenge of reproduction in the marine environment. In the depths where sunlight fades to blue, many corals produce their own ethereal light, shows through natural fluorescence, transforming the reef into an underwater aurora that rivals the northern lights in beauty. When excited by blue or ultraviolet light, fluorescent proteins in their tissues emit brilliant greens, reds, oranges, and pinks, creating a hidden dimension of the reef that remained unknown to science until recently. This fluorescence transforms the underwater landscape into something from another world, where familiar coral formations become alien sculptures glowing with internal light. The discovery of coral fluorescence has revolutionized our understanding of reef biology and opened new frontiers in medical research. This fluorescence isn't just beautiful, it serves important biological functions that scientists are still working to understand completely. Some fluorescent proteins may protect corals from harmful ultraviolet radiation, acting like biological sunscreen that converts damaging UV rays into harmless visible light. Others might enhance photosynthesis by converting blue light, which penetrates deeper into water, and into red light that their symbiotic algae can use more efficiently for energy production. The proteins act as biological transformers, taking light wavelengths that are abundant in the ocean and converting them to wavelengths that are more useful for biological processes. This light conversion may give fluorescent corals a competitive advantage in deeper waters where red light doesn't naturally penetrate. Different coral species produce different fluorescent signatures, like biological fingerprints written in light, and the patterns can vary even within a single colony based on environmental conditions and genetic factors. Some corals fluoresce only at their growing tips, where new polyps are actively building skeleton, creating glowing borders that outline their growth patterns, while others glow uniformly across their entire surface in sheets of colored light. The intensity and color of fluorescence can change with water temperature, nutrient availability, and even the time of day. Night divers, using special blue lights and yellow filters, can reveal this hidden world of biological illumination that exists alongside the familiar daytime reef, discovering a secret dimension that has existed for millions of years but remained invisible to human eyes until the development of appropriate technology. The fluorescent proteins in corals have revolutionized medical and biological research in ways that no one could have predicted when they were first discovered. Green fluorescent protein, originally discovered in jellyfish but found in many corals in various forms, has become an essential tool for scientists studying cellular processes at the molecular level. These proteins allow researchers to track individual cells through development, study gene expression in real time, and visualize biological processes in living organisms with unprecedented clarity. Scientists have used coral fluorescent proteins to watch cancer cells spread, observe how memories form in brain neurons, and track the development of organs in embryos. The Nobel Prize in Chemistry was awarded for the discovery and development of green fluorescent protein, highlighting the profound impact that coral fluorescence has had on modern science. Swimming over a fluorescent coral reef at night is like drifting through a living constellation, each coral colony glowing with its own internal light, revealing beauty that exists whether or not anyone is there to witness it. The intensity and colors of fluorescence can change with coral health, environmental conditions, and even time of day, creating a dynamic light display that reflects the hidden biochemistry of reef life. Stressed corals may increase their fluorescence as a protective response, creating brilliant displays that are both beautiful and alarming. Some scientists believe that fluorescent patterns may serve as communication signals between coral colonies, allowing them to share information about environmental conditions or as attractants for specific types of marine life that can see these fluorescent wavelengths. The discovery of fluorescent corals has opened new avenues of research into the functions of these remarkable proteins and their evolutionary origins. Some fluorescent proteins appear to be ancient evolutionary adaptations that have been conserved across millions of years, suggesting they provide important survival advantages that have been maintained through countless generations. Others may be more recent innovations that help corals adapt to specific environmental conditions in their local habitats. The diversity of fluorescent proteins in corals suggests that this trait has evolved independently multiple times, with different coral lineages developing their own unique solutions to similar environmental challenges. Research into coral fluorescence has revealed that the proteins responsible for these light shows are incredibly diverse, with each species potentially producing unique variants. Scientists have identified dozens of different fluorescent proteins in corals, each with unique spectral properties and potential applications in biotechnology and medicine. Some glow green, others red, orange, or even far-red wavelengths that are invisible to human eyes, but may be visible to reef fish. The molecular mechanisms underlying coral fluorescence involve complex protein structures that absorb light at one wavelength and emit it at another through a process called Stokes shift. These proteins can be incredibly efficient, converting nearly 100% of absorbed light energy into fluorescent emission with minimal heat generation. Fluorescent proteins in corals are often linked to stress responses, with some species increasing fluorescence when exposed to environmental challenges such as temperature changes, pollution, or physical damage. This suggests that fluorescence may serve as a visible indicator of coral health and stress levels, potentially providing an early warning system for reef managers. Some corals produce particularly bright fluorescence just before bleaching, creating a final burst of color before expelling their symbiotic algae. The patterns of fluorescence in coral colonies can be incredibly complex, with different regions of the same colony producing different colors and intensities of light based on local conditions. These patterns may reflect the physiological state of different parts of the colony, the density of symbiotic algae, or the local environmental conditions they experience, such as light exposure or water flow patterns. The stability of coral fluorescent proteins under extreme conditions has made them valuable for scientific and commercial applications. Some coral proteins remain fluorescent at temperatures that would destroy other biological molecules. In pH conditions that would denature most proteins, and even after being dried and rehydrated. This remarkable stability makes them ideal for long-term experiments, industrial applications, and even consumer products like fluorescent aquarium decorations that mimic natural coral fluorescence. Coral reefs serve as natural seawalls, absorbing and dissipating wave energy before it reaches shore through sophisticated physical mechanisms that engineers are only beginning to fully appreciate. Healthy coral reefs can reduce wave energy by 70–90%, protecting coastal communities from storm surge, erosion, and flooding with an effectiveness that rivals or exceeds human-engineered structures. This protective service is worth billions of dollars globally and becomes increasingly important as sea levels rise and storms intensify due to climate change. Without coral reefs, many tropical coastlines would be uninhabitable, constantly battered by waves that would erode beaches, contaminate freshwater supplies with salt, and destroy coastal infrastructure. The three-dimensional structure of coral reefs is perfectly designed to break up waves through a combination of physical processes that work together to dissipate energy. As waves encounter the reef, they trip over the coral formations, losing energy through turbulence and friction in a cascade of energy transformation. The rough surface of coral colonies creates drag that slows water movement, while the porous structure of the reef framework allows water to flow through countless channels, further dissipating wave power. The more complex the reef structure, the more effectively it dissipates wave power through these multiple mechanisms. Branching corals create particularly effective wave barriers, their intricate shapes forcing water to navigate countless obstacles, splitting wave energy into smaller and smaller components until it is effectively neutralized. The reef acts like a massive shock absorber, converting the organized energy of waves into disorganized, turbulent motion that poses no threat to shorelines. During major storms, coral reefs can mean the difference between minor coastal damage and catastrophic destruction that displaces entire communities. Islands and coastlines protected by healthy reefs suffer significantly less erosion and flooding than areas where reefs have been damaged or destroyed by pollution, overfishing, or development. This protection extends inland for miles, as reefs prevent the kind of deep wave penetration that can cause groundwater contamination, destroy agricultural lands, and undermine the foundations of buildings. Historical records show that areas with healthy coral reefs have survived hurricanes and tsunamis that devastated nearby unprotected coastlines, demonstrating the life-saving value of these natural barriers. The 2004 Indian Ocean tsunami provided tragic evidence of this protection areas, with intact coral reefs experienced significantly less damage and loss of life than areas where reefs had been destroyed. Unfortunately, many coastal communities are only beginning to recognize this service, as reef degradation increases their vulnerability to storms and everyday wave action. The economic value of reef protection often exceeds the cost of reef conservation by orders of magnitude, making healthy reefs one of the most cost-effective forms of coastal defense available. Studies have shown that maintaining coral reefs costs a fraction of building and maintaining seawalls or other artificial coastal defenses, while providing additional benefits like tourism, fisheries, and biodiversity conservation. Every meter of reef lost to human activities requires millions of dollars in artificial structures to replace its protective function, and these artificial structures lack the self-repairing capabilities of living reefs. Every healthy coral reef is essentially a living seawall, built and maintained by millions of tiny animals, working to protect not only themselves, but also the human communities that depend on calm, protected waters. The wave protection provided by reefs allows the development of beaches, mangrove forests, and seagrass beds in the calmer waters behind the reef, creating additional coastal protection through ecosystem layering that amplifies the protective effect. As climate change increases storm intensity and frequency, the protective value of coral reefs becomes even more crucial for coastal resilience, potentially determining which communities survive and which must be abandoned to the rising seas. Engineering studies have shown that coral reefs are remarkably efficient at wave energy dissipation, outperforming many human-designed structures. The combination of their height, relative to water depth, surface roughness from coral colonies, and porosity of the reef framework creates optimal conditions for breaking up wave energy across multiple scales. The reef crest, where waves first encounter shallow coral formations, can dissipate up to 86% of wave energy through breaking, while the reef flat behind continues to reduce wave height through friction. Attempts to replicate this protection with artificial structures often prove less effective and far more expensive than maintaining healthy natural reefs, while also lacking the aesthetic and ecological benefits of living coral systems. The protective function of reefs varies with their health and structural complexity, creating a direct link between ecosystem conservation and human safety. Degraded reefs with reduced coral cover and simplified structure provide significantly less wave protection, demonstrating the direct connection between ecosystem health and human safety. This connection makes coral conservation a matter of coastal defense and economic security, as well as environmental protection. The loss of just 1 meter of reef height can double or triple the wave energy, reaching shore, dramatically increasing erosion rates and storm damage potential. The mechanisms of wave energy dissipation by coral reefs involve multiple physical processes working in concert. Wave breaking on the reef crest creates turbulence that dissipates energy through the conversion of organized wave motion into chaotic water movement. Friction along the rough coral surface further reduces wave power through boundary layer effects, while the complex three-dimensional structure creates multiple opportunities for energy loss as waves navigate through coral formations. The porosity of the reef framework allows water to flow through numerous channels, creating additional friction and turbulence that removes energy from the wave system. These processes work together in ways that are difficult to replicate with artificial structures, making natural reefs irreplaceable for coastal protection. Different types of coral formations provide different levels and styles of coastal protection, each suited to particular coastal environments. Barrier reefs that parallel the coastline provide broad-scale wave protection across entire coastal regions, while fringing reefs that grow directly from shore offer more localized but intense protection. Patch reefs and coral gardens can provide intermediate levels of protection in areas where larger reef structures don't develop, creating stepping stones of wave reduction that collectively protect coastlines. The spacing and arrangement of reef formations create wave interference patterns that can either amplify or reduce wave energy at specific locations, requiring careful consideration in coastal management planning. When water temperatures rise too high, corals expel their colorful zooxanthellae algae in a stress response called bleaching, revealing the white limestone skeleton beneath, in a transformation that can occur in just days or weeks. While severe bleaching can kill corals, this dramatic response is actually a survival strategy that can allow corals to endure temporary environmental stress by jettisoning partners that have become liabilities. The bleaching response represents millions of years of evolution, a last resort mechanism that allows corals to survive conditions that would otherwise be fatal. When corals detect that their algae are producing harmful levels of reactive oxygen species due to thermal stress, they actively expel these formerly beneficial partners to prevent cellular damage that could kill the entire colony. Bleaching gives corals a chance to reset their symbiotic relationships, like reformatting a computer to eliminate corrupted files. By expelling algae that may be producing harmful oxygen radicals in high-temperature conditions, corals can sometimes take up new, more heat-tolerant strains of zooxanthellae when conditions improve. This flexibility has helped corals survive past climate fluctuations over geological timescales and may be crucial for their adaptation to current warming trends. The process is remarkably sophisticated. Corals don't simply expel all algae indiscriminately, but can selectively release specific strains while retaining others that may be more stress-tolerant. This selective expulsion suggests an active management system, where corals monitor the performance of their algal partners and make decisions about which to keep and which to release. Recovery from bleaching requires time and stable conditions that allow corals to rebuild their symbiotic partnerships. Mildly bleached corals may recover within weeks if temperatures drop quickly and remain stable, while severely bleached colonies may need months or even years to rebuild their algae populations and regain their color. During recovery, corals must survive on stored energy reserves in their tissues and whatever food they can capture through feeding with their tentacles, making them more vulnerable to disease, predation, and other stresses. The recovery process follows predictable stages first. Surviving algae within the coral tissue begin to reproduce. Then, new algae may be acquired from the water column. Finally, the algal population density returns to normal levels, and the coral regains its characteristic coloration. Some coral species, and even some individual colonies, show greater bleaching resistance than others, demonstrating remarkable variation in thermal tolerance. Scientists are studying these resilient corals to understand how they maintain their algae partnerships under stress, searching for the genetic and physiological traits that confer resistance. This research may help identify coral populations that could seed reef restoration efforts or naturally adapt to warming oceans through natural selection. Some corals have been found to harbor multiple types of algae simultaneously, allowing them to shift their dominant symbiont type in response to changing conditions, a phenomenon called symbiont shuffling that may be key to coral survival in a warming world. A bleached coral reef is not necessarily a dead reef. It is a reef in crisis, but with potential for recovery. Under the stark white limestone lies a living animal, temporarily separated from its partners, but potentially capable of remarkable recovery when conditions align. The bleaching response appears to be an ancient adaptation that has allowed corals to survive temperature fluctuations over millions of years, though the current rate and intensity of warming may exceed their adaptive capacity. Some corals can survive multiple bleaching events, building thermal tolerance through repeated exposure to moderate stress, in a process similar to athletic conditioning. Each survival event may select for more resistant individuals, potentially leading to evolution of heat-tolerant populations. The bleaching process itself is complex and not fully understood despite decades of research. Corals don't simply expel all their algae at once. The process often begins with the loss of specific algae types, while others remain, creating a patchwork of bleached and unbleached tissue. This selective expulsion suggests that corals have sophisticated mechanisms for monitoring and controlling their algal populations. The cellular machinery involved includes specialized proteins that can detect stress in algal cells, signaling pathways that trigger expulsion, and transport systems that physically remove algae from coral tissue. Understanding these mechanisms is crucial for predicting which corals will bleach and which might resist. Recovery from bleaching involves more than just taking up new algae. It requires comprehensive cellular repair and metabolic adjustment. Corals must repair cellular damage caused by oxidative stress, rebuild their tissue architecture that may have been compromised during bleaching, and restore normal metabolic function, including the complex nutrient exchange systems with their algal partners. This process requires significant energy investment at a time when the coral's primary energy source, photosynthesis by algae, is reduced or absent. Corals may lose significant tissue during bleaching, and recovery includes not just re-establishing algal populations, but also regenerating lost tissue and rebuilding energy reserves. The molecular mechanisms of bleaching involve complex stress responses at multiple biological levels. High temperatures can cause the photosynthetic machinery of zooxanthellae to malfunction, producing harmful reactive oxygen species that damage both algal and coral cells. The coral's response involves heat shock proteins that attempt to protect cellular machinery, antioxidant systems that neutralize reactive oxygen species, and ultimately, the expulsion mechanism that removes damaged algae. Research has revealed that some corals produce special protective proteins during thermal stress, including fluorescent proteins that may shield remaining algae from excessive light, and mycosporin-like amino acids that act as natural sunscreens. The factors that influence bleaching recovery include not just temperature, but also water quality, light levels, nutrients, and the presence of diseases or other stressors. Clean water with low sediment and pollution levels promotes faster recovery, while high nutrient levels may actually impede recovery by promoting algal growth that competes with corals. Understanding these factors is crucial for predicting which corals are most likely to survive bleaching events, and for developing strategies to enhance recovery through active management interventions. The relationship between coral reefs and fish goes far beyond simple habitat provision, extending into complex metabolic partnerships that sustain entire ecosystems. Fish play crucial roles in coral nutrition, growth, and health through a complex web of interactions that scientists are still discovering after decades of research. The nutrients that fish provide to corals are not just supplements, but essential components of coral metabolism that can determine the difference between thriving reefs and struggling colonies. From tiny cleaner fish that remove parasites to large schools that provide nutrients through their waste, fish are essential partners in reef ecosystems, creating nutrient cycles that transform nutrient-poor tropical waters into some of the most productive ecosystems on Earth. Many fish species directly feed corals through their excretions, providing a constant stream of bioavailable nutrients in waters where such resources are vanishingly scarce. Fish digest algae, plankton, and other organic matter, then release dissolved nutrients like nitrogen and phosphorus that corals absorb directly through their tissues. These nutrients arrive in forms that corals can immediately use: ammonia, urea, and phosphate, requiring no additional processing. Large schools of fish can provide substantial fertilization for coral colonies, particularly in nutrient-poor tropical waters, where these elements are scarce and limit growth. A single school of fish sheltering in a coral formation at night can increase local nutrient concentrations by several orders of magnitude, creating hotspots of productivity that support enhanced coral growth and reproduction. Some fish actively tend to corals, like underwater gardeners, their behaviors directly influencing coral health and growth rates. Damselfish cultivate algae gardens on dead coral surfaces, defending their territories aggressively and inadvertently protecting nearby healthy corals from algae overgrowth and coral predators. Their constant activity stirs up sediment and detritus, preventing it from smothering coral polyps. Parrotfish create sandy areas where coral larvae can settle by scraping algae from hard surfaces with their powerful beaks, while their constant grazing prevents algae from smothering coral colonies. The sound of parrotfish feeding, a constant crunching audible throughout the reef, is the sound of ecosystem maintenance in action. Without these herbivorous fish, many reefs would be overwhelmed by algae within months. Cleaner fish provide another essential service, removing dead tissue, parasites, and bacterial infections from coral colonies with surgical precision. Specialized cleaning stations often develop around large coral formations, where fish wait in organized queues for services from cleaner wrasses and gobies. These tiny fish pick off parasites, remove dead scales, and even clean wounds, providing health services that would cost millions if replicated by human intervention. This relationship helps keep corals healthy and disease-free, while providing the cleaners with a reliable food source. The trust required for predatory fish to allow small cleaners into their mouths demonstrates the sophisticated social contracts that govern reef ecosystems. The loss of fish populations can directly impact coral health in cascading effects that transform entire reef systems. Overfishing of herbivorous fish often leads to algae overgrowth that can smother corals within a single season, while the removal of cleaning fish may increase disease rates in coral colonies. The nutrients provided by fish waste can be so important that reefs with depleted fish populations show slower coral growth rates, reduced reproduction success, and increased susceptibility to bleaching. This creates a feedback loop where coral health depends on fish populations, while fish populations depend on coral habitat, a reciprocal relationship that has evolved over millions of years. Fish also contribute to coral nutrition through their role in moving nutrients between different parts of the reef, and even between reef and open ocean. Schools of fish that feed in open water during the day and shelter in the reef at night import nutrients from the pelagic zone, essentially subsidizing the reef ecosystem with resources from elsewhere. Other fish species feed in seagrass beds or sandy areas and excrete in coral zones, creating nutrient transport networks that help distribute essential elements across the reef system. This biological conveyor belt of nutrients is particularly important for corals growing in nutrient-poor environments, where local resources alone would be insufficient for growth. Some fish species have co-evolved with specific coral species, forming partnerships so intimate that neither can thrive without the other. Certain gobies, for example, live exclusively within the branches of particular coral species, defending their coral hosts from predators and parasites, while receiving protection and feeding opportunities in return. These gobies will attack much larger fish that attempt to feed on their coral hosts, demonstrating remarkable courage in defense of their homes. The presence of these guardian fish can increase coral growth rates by up to 30% compared to undefended colonies. The behavioral interactions between fish and corals can be remarkably sophisticated, involving recognition, memory, and decision-making. Some fish species appear to recognize individual coral colonies and modify their behavior based on the health and condition of their coral partners. Fish may preferentially excrete near stressed corals that need additional nutrients, or increase their cleaning activities when corals show signs of disease. This suggests a level of ecological awareness that was previously unrecognized and challenges our understanding of fish cognition. Fish swimming patterns around coral colonies create water currents that enhance coral feeding and waste removal. The constant movement of fish creates mixing that brings fresh seawater into contact with coral tissues, delivering oxygen and food particles while removing metabolic waste. Large schools of fish can create significant water flow through their collective movement, essentially providing free water circulation services that would otherwise require wave action or currents. This bioturbation is particularly important in protected reef areas where water movement might otherwise be limited. The chemical communication between fish and corals involves complex molecular signals that coordinate their interactions. Corals may release chemical cues that attract beneficial fish species while deterring potential threats, creating chemical gradients that guide fish behavior. Some fish can detect stress hormones released by corals and respond by increasing their nutrient provisioning or protective behaviors. This chemical dialogue represents an ancient language that predates human speech by hundreds of millions of years. Despite their peaceful appearance, corals engage in constant territorial warfare, using sophisticated chemical weapons that rival the complexity of modern military arsenals. When different coral colonies grow close to each other, they often engage in coral wars, slow-motion battles for space and resources that can last for years and determine which species dominate different areas of the reef. These battles shape reef architecture and biodiversity, creating complex mosaics of species distribution that reflect ongoing and historical conflicts. The weapons deployed in these battles include toxins, digestive enzymes, and growth inhibitors that have evolved over millions of years of evolutionary arms races between competing species. Many corals produce sweeper tentacles, specialized defensive structures that can extend several times the length of normal feeding tentacles, sometimes reaching lengths of 15 to 20 centimeters. These weapons are loaded with concentrated stinging cells and digestive enzymes that can kill competing corals on contact. The development of sweeper tentacles is triggered by chemical detection of nearby competitors, taking several weeks to fully form. When two coral colonies meet, the aggressor species may deploy sweeper tentacles at night to digest and kill parts of its neighbor, claiming the newly available space through cellular destruction. The attacked coral may respond by developing its own sweeper tentacles, leading to prolonged border conflicts that can continue for years. These battles occur in slow motion by human standards, but represent intense competition for the limited space available on reef surfaces. Some corals use chemical warfare at a distance, releasing allelopathic compounds into the water that inhibit the growth or settlement of competing species without requiring physical contact. These chemical weapons can create dead zones around aggressive coral species, where other corals cannot establish themselves, giving the chemical warriors more space to grow uncontested. The compounds involved include terpenoids, which interfere with cellular metabolism, and various proteins that disrupt normal physiological processes in target species. Some species can detect these chemical gradients and avoid settling in areas where they would be at a competitive disadvantage, creating spatial patterns that reflect invisible chemical landscapes. The hierarchy of coral aggression is complex and species-specific, forming what scientists call competitive networks. Fast-growing branching corals often lose chemical battles to slower-growing massive species with more potent defensive systems, creating a rock-paper-scissors dynamic where no single species dominates all interactions. However, the branching corals may win through rapid growth, shading their chemical superiors and claiming the valuable upper reef areas where light is most abundant. This multidimensional competition creates diverse reef communities where different strategies can coexist, each successful in particular contexts. Environmental conditions can shift competitive advantages, with some species becoming more aggressive during stressful periods, while others lose their chemical defenses. These invisible battles shape the structure and composition of entire reef systems in ways that are only visible through time-lapse observation. Walking over a coral reef, you are observing the frozen moment of countless ongoing territorial disputes, each coral colony, both weapon and fortress, in an ancient war for living space. The chemical warfare between corals creates a complex mosaic of species distributions, with aggressive species dominating prime real estate, while less combative species find refuge in marginal areas or develop strategies to coexist with their aggressive neighbours. Some corals have evolved tolerance to specific chemical weapons, allowing them to live closer to aggressive neighbours than other species can manage, creating networks of resistance and susceptibility that influence community assembly. The weapons used in coral warfare are incredibly diverse, representing millions of years of evolutionary innovation. Some species produce cytotoxins that directly kill competitor tissue through cellular disruption, while others release growth inhibitors that slow down neighbouring colonies without killing them outright. Certain corals even produce compounds that interfere with the reproduction of nearby species, preventing them from successfully spawning or recruiting new individuals to maintain their populations. Some release enzymes that break down the protective mucus layers of competitors, making them vulnerable to infection and predation. The diversity of chemical weapons suggests multiple independent evolutionary origins of competitive mechanisms. These chemical battles can be observed in real time with careful study and specialized imaging techniques. Time-lapse photography reveals the slow-motion advance and retreat of coral borders as species gain and lose territory in response to chemical attacks and defenses. Fluorescent imaging can show the damage caused by chemical weapons before it becomes visible to the naked eye, revealing zones of stressed tissue surrounding competitive interactions. The outcomes of these battles can shift based on environmental conditions, with some species gaining chemical advantages during stress periods, while others become more vulnerable to attack. The evolutionary arms race between coral species has led to increasingly sophisticated chemical defenses and counter-defenses that continue to evolve. Some corals have evolved the ability to detoxify specific compounds produced by their neighbours through specialized enzymes, while others have developed new toxins that overcome existing resistance mechanisms. This ongoing evolution creates a dynamic, competitive landscape where advantages are temporary and species must constantly adapt to maintain their position. The rate of evolution in these chemical systems may be accelerated by environmental stress, potentially leading to rapid changes in competitive hierarchies. Environmental factors such as water flow, temperature, and nutrient availability can dramatically influence the outcomes of coral chemical warfare. Strong currents may dilute chemical weapons, reducing their effectiveness and allowing weaker competitors to coexist with aggressive species, while stagnant conditions can allow toxins to accumulate to lethal concentrations, intensifying competitive interactions. Temperature stress may reduce the production of chemical defenses in some species, while enhancing it in others, potentially reversing competitive hierarchies during bleaching events. Nutrient enrichment can alter the production of allelopathic compounds, with some species becoming more aggressive under high nutrient conditions. The study of coral chemical warfare has revealed that many of the compounds used as weapons also serve other functions, demonstrating the efficiency of evolution. Some defensive chemicals also deter predators, prevent fouling by algae and bacteria, or protect against UV radiation. This multifunctionality makes coral chemical systems particularly complex and interesting to researchers studying natural products chemistry. Many coral-derived compounds have shown promise as pharmaceuticals, including anti-cancer agents and antibiotics, highlighting the practical value of understanding coral chemical warfare. While most corals appear permanently attached to the reef, some species are capable of limited movement, especially during their juvenile stages, challenging our fundamental assumptions about these organisms. Young coral colonies can slide along hard surfaces using coordinated tissue contractions, rotate to face favorable conditions by redistributing their weight, or even detach and relocate to better sites when conditions become unfavorable. This mobility, though slow by human standards, sometimes just millimeters per day, can be crucial for coral survival and distribution across reef systems. The discovery of coral mobility has revolutionized our understanding of reef dynamics, revealing that these seemingly sessile animals are more active participants in their environment than previously imagined. Mushroom corals are among the most mobile, with some species capable of moving several meters over the course of months through a variety of mechanisms. They use tissue inflation and deflation to rock back and forth, gradually working their way up slopes or away from unfavorable conditions like a slow-motion inchworm. The process involves coordinated inflation of tissue on one side while the other side contracts, creating a rocking motion that gradually shifts the coral's position. Some can even right themselves if flipped over by strong currents or storms, using a combination of tissue expansion and mucus secretion to create leverage against the substrate. This self-righting ability can mean the difference between survival and death, as inverted corals cannot feed properly and may be smothered by sediment. Small coral fragments can also exhibit surprising mobility that plays important roles in reef recovery and expansion. When pieces break off from parent colonies during storms, they may continue moving until they find suitable attachment sites, sometimes traveling several meters across the reef floor. This natural fragmentation and movement helps corals colonize new areas and recover from damage more quickly than previously thought possible. The fragments can sense environmental gradients, light, water flow, chemical cues, and orient their movement toward favorable conditions. Some fragments can even delay permanent attachment while searching for optimal settlement sites, maintaining mobility for weeks or months while exploring their options. The movement isn't random. Corals generally move toward better light conditions, away from sediment accumulation, or toward more favorable water flow patterns with remarkable consistency. Some species can sense these gradients through specialized cells that detect light intensity, water chemistry, and physical factors, and actively orient themselves to optimize their growth conditions. This demonstrates a level of environmental awareness that challenges our assumptions about these seemingly passive animals. Research has shown that mobile corals can detect and move away from aggressive neighbors, avoid areas with high sedimentation, and position themselves in locations with optimal water flow for feeding. This behavioral sophistication suggests that corals process environmental information and make decisions about movement, though the mechanisms remain poorly understood. Even the slowest movement can be significant over geological time, accumulating into substantial relocations. Coral colonies that move just centimeters per year can travel considerable distances over their multi-century lifespans, potentially escaping local environmental problems or finding new territory for expansion. A coral that moves just 1 centimeter per year could travel 10 meters over a 1,000-year lifespan, enough to completely change its position on the reef. The mechanisms of coral movement involve complex interactions between tissue expansion, water uptake, and secretion of slippery mucus that reduces friction with the substrate. Some species produce specialized mucus with lubricating properties that allow them to slide more easily across surfaces, while others use changes in buoyancy to lift themselves slightly off the substrate during movement. Coral movement is most pronounced in species that live on sandy or rocky substrates, rather than those that grow directly on reef frameworks. Free-living corals must be able to escape burial by sediment and position themselves in optimal light conditions, making mobility a crucial survival skill that has evolved independently in multiple coral lineages. These mobile species often have flattened or rounded shapes that facilitate movement, unlike the branching or encrusting growth forms of sessile species. The ability to move allows these corals to exploit habitats that would be unsuitable for permanently attached species, expanding the range of environments where corals can survive. The study of coral movement has revealed that these animals are far more dynamic than previously understood, actively shaping their environment rather than passively responding to it. Rather than being fixed in place for their entire lives, many corals actively adjust their positions in response to changing environmental conditions, demonstrating a form of behavioral adaptation that was previously unrecognized in these ancient animals. Time-lapse photography has captured corals moving in response to seasonal changes in light angles, storm damage, and competitive interactions with other organisms. Some corals show daily movement patterns, adjusting their position slightly in response to the sun's path across the sky. Some coral species use their mobility to escape from predators or parasites with surprising effectiveness. When attacked by crown-of-thorns, starfish, or other coral predators, mobile corals may be able to move away from danger, while sessile species must rely entirely on defensive chemicals or structural barriers. The speed of escape may be slow, perhaps a few centimeters per day, but it can be sufficient to avoid slow-moving predators or to move out of territories patrolled by coral-eating fish. This mobility advantage may explain why some free-living coral species persist in areas with high predation pressure where attached species cannot survive. Environmental factors such as temperature, light, and water flow can influence coral movement patterns in complex ways. Corals may move more actively during certain seasons when environmental conditions are changing, or in response to specific environmental cues like storm approach. Some species show increased movement during reproductive seasons, possibly to optimize positions for spawning success. Temperature changes can affect the viscosity of lubricating mucus, potentially influencing movement rates. Understanding these environmental influences is crucial for predicting how coral communities might reorganize in response to climate change. Large coral reef systems actually influence local weather and climate patterns through their effects on ocean temperature, humidity, and wind patterns, functioning as massive climate modification engines. The shallow waters above coral reefs heat up during the day and cool at night differently than deeper ocean waters, creating temperature gradients that can drive local wind systems and cloud formation with the power of natural atmospheric engines. This thermal difference can be substantial. Shallow reef waters may be several degrees warmer than surrounding deep water during the day, creating convection currents that extend high into the atmosphere. These temperature variations create pressure differences that generate local wind patterns, which in turn affect cloud formation, precipitation, and even the paths of larger weather systems. The Great Barrier Reef, extending over 2,300 kilometers along the Australian coast, significantly influences regional climate patterns in ways that affect weather hundreds of kilometers inland. During the day, the reef warms the water above it, creating rising air currents that can trigger cloud formation and afternoon thunderstorms that provide crucial rainfall to coastal areas. These localized weather patterns help distribute rainfall across nearby coastal areas that might otherwise be much drier. The reef acts like a massive solar collector, absorbing heat during the day and releasing it at night, creating a thermal engine that drives atmospheric circulation. Scientists have documented that rainfall patterns along the Queensland coast correlate with the health and extent of the Great Barrier Reef, suggesting that reef degradation could alter regional precipitation patterns. Coral reefs also influence ocean circulation patterns through their effects on wave action and tidal flows, creating complex hydrodynamic effects that extend far beyond the reef itself. The complex three-dimensional structure of reefs creates turbulence and mixing that affects how water masses move across reef systems, influencing nutrient distribution and temperature patterns. This mixing can bring nutrient-rich, deeper water to the surface through upwelling, supporting productivity in surrounding waters, and affect the distribution of marine life across the broader region. The reef structure modifies tidal flows, creating jets and eddies that can transport larvae, nutrients, and heat over large distances. These circulation patterns can influence the movement of entire water masses, affecting ocean currents that play crucial roles in global climate regulation. The albedo effect of coral reefs, their ability to reflect sunlight, also influences local temperatures in measurable ways. Healthy coral reefs, with their bright colors and complex surfaces, reflect more sunlight than would bare sediment or algae-covered surfaces, helping to keep local waters cooler than they would otherwise be. This reflection can reduce local water temperatures by up to 1 degree Celsius, which may seem small, but can be crucial for coral survival during heat stress events. The bleaching of corals changes their reflective properties, potentially creating feedback loops where bleached reefs absorb more heat, making recovery more difficult. Different coral species and growth forms have different reflective properties, creating complex patterns of heating and cooling across reef systems. As coral reefs decline due to climate change and other stresses, their ability to influence local weather patterns may also be compromised, potentially creating feedback loops that accelerate environmental changes in reef regions. The loss of reef structure can alter wave patterns, affecting coastal erosion and the distribution of sediments that influence water clarity and temperature. Degraded reefs may absorb more solar radiation, increasing local water temperatures and potentially affecting the formation of clouds and precipitation. These changes could have cascading effects on coastal agriculture, water resources, and human settlements that depend on reef-modified weather patterns. The weather-modifying effects of coral reefs extend beyond their immediate vicinity through complex atmospheric interactions. Large reef systems can influence atmospheric circulation patterns that affect weather hundreds of kilometers away, creating regional climate effects that are only beginning to be understood. The evaporation of water from shallow reef areas contributes significantly to regional humidity and precipitation patterns, with some estimates suggesting that reef systems contribute up to 10% of atmospheric moisture in coastal regions. This moisture contribution is particularly important during dry seasons, when other sources of atmospheric water are limited. Research has shown that the loss of coral reefs can lead to measurable changes in local climate patterns that affect human communities and natural ecosystems. These changes include altered rainfall distribution, with some areas receiving more rain, while others experience drought, increased coastal temperatures that affect agriculture and human comfort, and changes in wind patterns that influence everything from sailing conditions to the dispersal of air pollution. Some studies suggest that healthy reef systems can reduce the intensity of tropical storms by disrupting the feedback mechanisms that allow these systems to strengthen. The complex topography of coral reefs creates microclimates that can vary dramatically over small distances, contributing to local biodiversity. Areas of shallow water may be several degrees warmer than adjacent deeper areas, creating temperature gradients that drive local air and water circulation patterns. These microclimates provide refuges for organisms during extreme weather events, allowing species to survive conditions that would be lethal in more homogeneous environments. The variety of microclimates within reef systems may be crucial for species adaptation to climate change, providing stepping stones for evolutionary adaptation. The biological activity of coral reefs also influences local weather through the release of gases and aerosols that can affect cloud formation and precipitation. Dimethyl sulfide, produced by reef organisms and their associated algae, may seed cloud formation, potentially increasing rainfall in reef regions, while other chemical emissions can influence atmospheric chemistry in ways that affect weather patterns. Some researchers hypothesize that healthy reefs produce more of these cloud-seeding compounds, creating a positive feedback loop where healthy reefs promote rainfall that reduces terrestrial runoff and maintains water quality. The loss of this biological weather modification could have far-reaching consequences for regional climate stability. Different types of coral reefs have different effects on local weather patterns, reflecting their size, structure, and position relative to coastlines. Barrier reefs that extend far from shore may have different climate effects than fringing reefs that grow close to land, with barrier reefs potentially creating larger-scale atmospheric effects while fringing reefs have more localized impacts. Atolls' ring-shaped reefs surrounding lagoons create unique weather patterns with calm conditions in the lagoon and more turbulent conditions on the ocean side, demonstrating how reef structure influences atmospheric conditions at multiple scales. Coral skeletons serve as detailed archives of past environmental conditions, recording information about ocean temperature, chemistry, and climate patterns with remarkable precision that rivals modern scientific instruments. By analyzing the chemical composition of coral cores, scientists can reconstruct climate conditions spanning centuries or even millennia, creating detailed records of how Earth's climate has changed over time with annual or even seasonal resolution. These natural archives provide irreplaceable data about climate variability before human industrial activity, establishing baselines for understanding current changes. The limestone skeletons of corals are like natural data loggers, continuously recording environmental conditions as they grow, layer by layer, year after year, creating libraries of information locked in stone. As corals grow, they incorporate trace elements from seawater into their calcium carbonate skeletons, with ratios that precisely reflect environmental conditions. The ratios of different elements and isotopes, strontium to calcium, magnesium to calcium, oxygen, isotopes, reflect the temperature and chemistry of the water when each layer of skeleton was formed. These chemical signatures are so reliable that scientists can determine past ocean temperatures to within 0.5 degree Celsius. Annual growth bands in coral skeletons, similar to tree rings but often more clearly defined, allow scientists to date these environmental records precisely, sometimes down to specific months or even weeks. The density of these bands varies with growth rate, which is influenced by temperature, light, and nutrient availability, providing multiple proxies for environmental reconstruction. Coral cores have revealed detailed information about past El Niño events, volcanic eruptions, and long-term climate cycles that would otherwise be lost to history. Some coral records extend back over 400 years, providing climate data from long before instrumental weather records began in most parts of the world. These records have been crucial for understanding natural climate variability and distinguishing human-caused climate changes from natural fluctuations that have always occurred. The coral archives have revealed that some climate oscillations operate on cycles of decades or centuries, patterns that would be invisible without these long-term records. They've shown how volcanic eruptions can cool global climate for years, how solar variations influence Earth's temperature, and how ocean currents have shifted over time. The precision of coral climate records is remarkable, exceeding what might be expected from biological systems. Scientists can detect temperature changes of less than 1 degree Celsius, identify specific years when major volcanic eruptions affected global climate, and even determine seasonal variations in temperature and rainfall from hundreds of years ago. Coral records have even revealed the timing and intensity of past hurricanes and tropical storms through disruptions in growth patterns and changes in isotopic composition. Storm events leave distinctive signatures in coral skeletons. Sudden changes in growth rate, incorporation of terrestrial materials, and shifts in chemical composition that mark these extreme events in the geological record. Living corals continue adding to these climate archives every day, creating real-time records of current ocean conditions that will be valuable for future scientists studying our era's climate changes. Today's corals are recording the rapid warming of the oceans, the changing chemistry caused by carbon dioxide absorption, and the increasing frequency of extreme weather events. The chemical signatures in coral skeletons can reveal information about past rainfall patterns through isotopes that reflect freshwater input, river discharge through terrigenous elements washed from land, pollution levels through heavy metal incorporation, and even solar activity through radiocarbon variations. This makes coral reefs among the most comprehensive paleoclimate archives available to science, providing multiple independent measures of past environmental conditions. The study of ancient coral reefs has also provided insights into how reef ecosystems have responded to past climate changes, offering crucial lessons for predicting future responses. By examining the species composition and growth patterns of fossil reefs, scientists can understand how coral communities adapted to previous warming and cooling periods, sea level changes, and ocean chemistry variations. These studies reveal that coral reefs have survived dramatic climate changes in the past, but that the rate of current change may exceed their adaptive capacity. Fossil reefs show evidence of community shifts, with different species assemblages dominating during different climate periods, suggesting that while reefs may persist, their composition could change dramatically. Coral climate records have been instrumental in understanding long-term climate cycles, such as the Pacific Decadal Oscillation and the Atlantic Multi-Decadal Oscillation that influence global weather patterns. These natural climate patterns operate on timescales of decades to centuries, making coral records essential for distinguishing natural variability from human-caused changes. Without coral records, scientists would have insufficient data to understand these long-term patterns that affect everything from hurricane frequency to drought cycles. The coral archives have revealed that these oscillations have varied in intensity and frequency over time, providing context for current climate variability. The techniques used to extract climate information from coral cores are constantly improving, allowing scientists to detect increasingly subtle environmental signals. New analytical methods, including laser ablation, mass spectrometry, microcomputed tomography, and synchrotron X-ray fluorescence, can reveal information about ancient ocean acidity through boron isotopes, nutrient levels through nitrogen isotopes, and even the activity of marine organisms through biomarkers preserved in the skeleton. These advancing techniques continue to unlock new information from coral cores collected decades ago, demonstrating that these archives become more valuable as analytical technology improves. Different coral species provide different types of climate information, with some being better indicators of temperature, while others are more sensitive to changes in ocean chemistry or biology. Massive porites corals are excellent temperature recorders due to their consistent growth and clear annual bands, while Diploria species better preserve hurricane signals. Understanding these differences is important for interpreting coral climate records accurately and for selecting appropriate species for different types of paleoclimate reconstruction. The geographic distribution of coral climate records allows scientists to reconstruct past climate patterns across entire ocean basins, revealing how climate change is propagated through the global system. While most corals depend on photosynthetic algae and require sunlight to thrive, deep water corals have evolved to live in the eternal darkness of the deep ocean, demonstrating remarkable adaptability. These cold water corals form extensive reef systems at depths where no sunlight penetrates, sometimes as deep as 6,000 meters below the surface, relying entirely on capturing food from the water column. Their existence challenges our fundamental understanding of what coral reefs are and where they can exist, revealing that the coral reef phenomenon is not limited to sunny, tropical shallows, but represents a fundamental organizing principle of marine life that functions across diverse environments. These deep-sea architects build their cities in perpetual night, proving that the coral way of life transcends the boundaries we once thought absolute. Deep-sea coral reefs exist in oceans around the world, from the continental shelves off Norway, where water temperatures hover near freezing, to the deep slopes of tropical islands, where pressure would crush most surface organisms. Without zooxanthellae to provide energy through photosynthesis, these corals must be efficient hunters, using their tentacles to capture plankton, the marine snow, the constant rain of organic particles from surface waters, and other organic particles that drift down from the sunlit world above. They've evolved enlarged polyps with extended tentacles to maximize their food-capturing ability, sometimes with polyps 10 times larger than their tropical cousins. Their feeding tentacles are often more numerous and more densely packed with stinging cells, creating highly effective food capture surfaces that can extract nutrition from the sparse resources of the deep ocean. Some deep-water coral species form massive reef structures comparable to tropical coral reefs in size and complexity. The Lofelia reefs off northern Europe create complex three-dimensional habitats that support diverse communities of fish, crustaceans, and other deep-sea creatures. Over 1,300 species have been documented in these cold-water reefs. These cold-water reefs grow much more slowly than tropical reefs, sometimes just 4 to 25 millimeters per year, but can persist for thousands of years, accumulating into structures hundreds of meters tall. The Roost Reef off Norway extends for over 35 kilometers and rises 35 meters above the seafloor, rivaling tropical reefs in scale despite growing in near-freezing darkness. Some deep-water reefs are estimated to be over 8,000 years old, having begun growing when sea levels were much lower during the last ice age. Deep-water corals face unique challenges, including limited food availability that would starve most organisms, extreme pressure that can exceed 600 atmospheres, and near-freezing temperatures that slow all biological processes. However, they also avoid many of the stresses that affect shallow-water reefs, such as bleaching from temperature fluctuations, storm damage from hurricanes, and direct human impacts like coastal development and pollution. The stable conditions of the deep ocean, where temperature varies by less than a degree over centuries, provide a refuge from the rapid environmental changes affecting surface waters. This stability may make deep-water reefs increasingly important as climate refugia for marine biodiversity. The discovery of extensive deep-water coral reefs has revolutionized our understanding of coral biology and marine biodiversity, revealing adaptations we never knew existed. These hidden ecosystems demonstrate that coral reefs are not confined to shallow tropical waters, but represent a fundamental way of organizing marine life across the world's oceans from the tropics to the poles. Deep-sea corals may be particularly important as refuges for marine species, as shallow-water reefs face increasing environmental pressures from climate change and human activities. Some scientists estimate that deep-water coral reefs may cover an area comparable to or even exceeding that of tropical coral reefs, though most remain unmapped and unexplored. Deep-water corals have evolved remarkable adaptations to survive in their challenging environment, demonstrating the incredible plasticity of the coral body plan. Many species have enlarged polyps and extended tentacles to maximize their food capture capabilities in nutrient-sparse waters. Some have developed specialized feeding behaviors, such as creating water currents with coordinated ciliary action to bring more food particles within reach, or producing sticky mucus nets that trap marine snow. Their skeletons are often more densely constructed than tropical corals, providing strength against deep ocean currents while using less energy for rapid growth. Some species can detect the faintest water movements that signal approaching prey, using sensory capabilities that exceed those of their shallow water relatives. The isolation of deep-sea coral reefs has led to high levels of endemism, with many species found nowhere else on Earth. These unique communities represent evolutionary laboratories, where life has experimented with new forms and functions over millions of years, without the genetic exchange common in shallow waters. Some deep-water coral species are living fossils, having remained virtually unchanged for millions of years, while their shallow-water relatives evolved and diversified. Others represent entirely unique evolutionary lineages that diverged from other corals hundreds of millions of years ago. The biodiversity of deep-sea coral reefs can rival that of tropical reefs, despite the harsh environmental conditions and absence of photosynthetic primary production. The complex three-dimensional structure created by coral growth provides habitat for hundreds of species of fish, including commercially important species like rockfish and shrimp, crustaceans that have evolved bizarre adaptations to deep-sea life, and other organisms that exist nowhere else. Many deep-sea fish species use coral reefs as nursery grounds, demonstrating that these ecosystems provide critical habitat services even in the absence of sunlight. The slow growth rates of deep-sea corals make these ecosystems extremely vulnerable to damage from human activities such as bottom trawling and deep-sea mining. Recovery from disturbance can take decades or centuries, with some damage potentially being irreversible on human timescales. A single pass of a bottom trawl can destroy coral colonies that took thousands of years to grow, eliminating habitat for countless associated species. This vulnerability makes conservation of deep-sea coral reefs a critical priority, though their remote location and the expense of deep-sea research make protection challenging. In the complex social hierarchies of coral reefs, some fish species have evolved remarkable abilities to change sex in response to social and environmental conditions, demonstrating biological flexibility that challenges our understanding of sexual development. This flexibility allows them to maximize their reproductive success and maintain stable populations in the confined spaces of coral reef territories, where opportunities for finding mates may be limited. The ability to change sex, called sequential hermaphroditism, has evolved independently in at least 7 fish families, suggesting it provides significant adaptive advantages in reef environments. This phenomenon occurs in over 500 species of reef fish, making it one of the most common reproductive strategies in these ecosystems, far more prevalent than the fixed-sex system familiar to mammals. Clownfish, made famous by animated movies, are sequential hermaphrodites that begin life as males and can transform into females when circumstances require it, a process called protandry. In each anemone territory, a dominant breeding pair consists of a large female and a smaller male, with several juvenile males waiting in the background in a strict size-based hierarchy. If the female dies, the breeding male transforms into a female within weeks, undergoing dramatic physiological changes, and one of the juveniles becomes the new breeding male, moving up the social ladder. This transformation involves not just reproductive organs, but also behavior, hormones, and even brain structure, representing one of the most complete sex changes in the animal kingdom. The new female immediately begins producing eggs, while the promoted male develops enhanced coloration and defensive behaviors. Many wrasse species follow the opposite pattern protogyny, beginning as females and transforming into males when they grow large enough to defend territories against rivals. Large male wrasses maintain harems of females in prime coral reef locations, aggressively defending their territories against rival males with elaborate displays and occasional physical combat. If the dominant male dies or is removed, the largest female in his territory will change sex and take over his role, usually within days to weeks. This transformation involves developing brilliant coloration, growing larger fins, and completely restructuring reproductive organs from egg-producing ovaries to sperm-producing testes. The behavioral changes are equally dramatic. The new male immediately begins courting females and defending territory with behaviors it never exhibited as a female. This sex-changing ability helps maintain optimal population structures in the confined spaces of coral reefs, where territory is limited and valuable. It ensures that breeding opportunities are not wasted, and that territories continue to be occupied by reproductively active fish, even when environmental changes or predation disrupt established social structures. Without this flexibility, the death of a dominant individual could could leave entire territories without breeding activity until new individuals arrived from elsewhere. The system also ensures that the largest, most successful individuals become males, who can defend territories and mate with multiple females, maximizing the genetic contribution of the fittest individuals. The transformation involves dramatic changes in behavior, coloration, and reproductive anatomy that can occur over weeks or months, demonstrating the remarkable plasticity of life in coral reef ecosystems. The process is controlled by complex hormonal systems that respond to social cues such as the absence of dominant individuals, population density, changes in the local territory, and territorial availability. The trigger often involves changes in stress hormones and sex steroids, with social interactions directly influencing hormone production. Some species can even reverse their sex changes if social conditions change again, a phenomenon called bidirectional sex change, highlighting the dynamic nature of reef social systems. This reversibility has been documented in some goby species that can change sex in both directions multiple times throughout their lives. Sex change in reef fish is often accompanied by dramatic changes in coloration and behavior that signal the individual's new status. Terminal phase, males often develop brilliant colors, electric blues, vibrant yellows, and deep purples and elaborate fins that help them attract females and intimidate rivals. These physical transformations can be so dramatic that early researchers thought they were observing different species, leading to centuries of taxonomic confusion. The color changes involve not just pigments, but also structural modifications to scales that create iridescent effects. Behavioral changes include developing elaborate courtship displays, territorial patrol patterns, and aggressive responses to intruders that the fish never exhibited in its previous sex. The social systems that drive sex change can be remarkably complex, involving multiple levels of dominance and sophisticated assessment of social position. Some wrasse species maintain multi-level hierarchies with different types of males, territorial supermales and sneaker males, competing for breeding opportunities through different strategies. Females must constantly assess their position in the hierarchy and the stability of the dominant male to determine when sex change might be advantageous. The ability to change sex provides flexibility that allows these social systems to adapt to changing conditions while maintaining reproductive output. The triggers for sex change vary among species and can include social factors like the ratio of males to females, environmental conditions such as temperature and food availability, and internal physiological states, including age and size. Some species require specific size thresholds before sex change can occur, while others respond primarily to social absence. Understanding these triggers has revealed the sophisticated social awareness that many reef fish possess, including the ability to count individuals, assess relative sizes, and remember social interactions over extended periods. Different fish species have evolved different mechanisms for controlling sex change, involving various hormonal pathways and genetic switches that scientists are still working to understand. Some use temperature-dependent systems, similar to those in reptiles, while others rely entirely on social cues. The study of these mechanisms has provided insights into the fundamental biology of sex determination and the evolution of reproductive systems. The genes involved in sex change are often the same ones that control sexual development in other vertebrates, but they remain plastic throughout life, rather than becoming fixed during development. Corals produce a protective layer of mucus that serves multiple functions, acting as sunscreen, antibiotic, and food source for many reef creatures, in a remarkable example of biological multifunctionality. This slimy coating, often invisible to casual observers and dismissed as mere slime, represents one of the reef's most important but overlooked biological products. The mucus layer can account for up to 50% of a coral's daily energy expenditure, suggesting its critical importance for survival. This investment of resources produces a complex substance containing hundreds of different compounds, from antimicrobial peptides to UV-blocking molecules, creating a pharmacy and cafeteria combined into a single remarkable secretion that sustains entire communities of organisms. The mucus contains antimicrobial compounds that help corals fight off bacterial and fungal infections with effectiveness that rivals modern antibiotics. Some of these compounds are so effective that researchers are investigating their potential for developing new human medicines, particularly important as antibiotic resistance threatens current treatments. The mucus contains peptides that can destroy bacterial cell walls, proteins that interfere with viral replication, and molecules that prevent biofilm formation, the slimy layers that protect pathogenic bacteria from immune responses. The mucus also traps sediments and debris, preventing them from settling on coral tissues where they could block light or harbour harmful bacteria. This physical barrier function is combined with chemical defences, creating a multi-layered protective system that has evolved over millions of years. Many fish and invertebrates feed directly on coral mucus, which is rich in sugars, proteins, and lipids, essentially a complete meal package. Parrotfish, surgeonfish, and other species regularly graze on coral surfaces, consuming not only algae, but also the nutritious mucus layer that can provide up to 70% of their daily energy needs. This feeding relationship helps keep coral surfaces clean while providing food for herbivorous reef fish, creating a win-win situation where both parties benefit. The mucus is constantly renewed, with some corals producing up to 4.8 litres per square metre per day, creating a renewable resource that supports entire food webs. The composition of mucus varies with coral species, time of day, and environmental conditions, providing a diverse menu for reef consumers. Some creatures have developed specialized relationships with coral mucus, evolving specific adaptations to exploit this resource. Certain gobies and cardinalfish live permanently within the mucus layers of large coral heads, their bodies coated in a protective layer of coral slime that shields them from parasites and predators. These fish feed on the constant supply of nutrients, while gaining protection from predators that avoid the coral's stinging cells. These fish may help their coral hosts by consuming harmful bacteria and parasites that could cause disease, creating a mutualistic relationship where both species benefit. Some species have evolved immunity to coral toxins, specifically to access this mucus resource, demonstrating the evolutionary importance of this substance. During times of stress, corals may increase mucus production dramatically, sometimes by 30 to 50 times normal rates, creating visible slicks on the water surface. While this may seem like a sign of poor health, increased mucus production can actually be a protective response that helps corals survive difficult conditions by providing additional antimicrobial protection and removing accumulated toxins. The mucus can carry away harmful substances, including heavy metals and metabolic waste, and provide additional antimicrobial protection during vulnerable periods when corals are more susceptible to disease. This stress response has been fine-tuned over evolutionary time to maximize survival during challenging periods. The chemical composition of coral mucus is incredibly complex, containing hundreds of different compounds that scientists are still cataloging. Many of these substances have antimicrobial, antiviral, or antifungal properties that protect corals from disease, with mechanisms different from conventional antibiotics. The mucus also contains chemical signals, pheromones, and allelochemicals that may help corals communicate with each other and with other reef organisms. Some compounds appear to attract beneficial bacteria while repelling harmful ones, creating a selective filter that shapes the coral's microbiome. The mucus contains enzymes that break down pollutants, glycoproteins that bind viruses, and even compounds that may help corals cope with thermal stress. Research into coral mucus has revealed that different coral species produce mucus with different properties tailored to their specific ecological needs. Some species produce mucus that is particularly effective against specific types of pathogens prevalent in their habitat, while others create mucus that is especially nutritious for associated organisms that provide protection services. Fast-growing branching corals often produce thinner, more watery mucus that is easily shed, while massive corals produce thicker, more persistent layers that provide longer-lasting protection. These differences reflect evolutionary adaptations to different ecological strategies and environmental challenges. The production of mucus requires significant energy investment by corals, with some species allocating up to half their daily energy budget to mucus production. This substantial cost suggests that the benefits must be equally substantial for the trait to have evolved and persisted. The multiple functions of mucus protection from pathogens, removal of sediment, provision of food for symbionts, facilitation of gas exchange, communication with other organisms, and protection from UV radiation make it one of the most important coral secretions for reef ecosystem function. The microbial communities associated with coral mucus are diverse and play crucial roles in coral health and disease resistance. The mucus layer hosts billions of bacteria per square centimetre, many of which produce antibiotics that protect their coral host. Understanding these microbial partnerships is important for coral conservation and reef health management, particularly as climate change and pollution disrupt these delicate relationships. The mucus microbiome can shift rapidly in response to environmental changes, potentially providing early warning signs of coral stress before visible symptoms appear. The white sand beaches that grace tropical islands are largely the product of parrotfish digestion, a fact that transforms how we view these iconic landscapes. These colourful reef fish spend their days crunching on coral with their powerful beak-like teeth, extracting algae and other organic matter, while inevitably consuming large quantities of coral skeleton along with their meals. The sound of their feeding, a constant grinding and crunching audible underwater, is the sound of beach creation in progress. This process, called bioerosion, is one of the most important geological forces in tropical coastal areas, literally creating the land on which millions of people live and vacation. Without parrotfish, many tropical islands would lack the beaches that define them, support their economies, and protect their shores from wave action. A single large parrotfish can produce over 200 pounds of sand per year through this feeding process, with some estimates reaching as high as 840 pounds for the largest species. The coral skeleton passes through their digestive system, where acid breaks down the calcium carbonate into fine particles that are excreted as sand. Their specialized digestive system includes a pharyngeal mill, essentially a second set of grinding teeth in their throat, that pulverizes coral fragments with forces that can exceed 530 pounds per square inch. The constant grinding action of parrotfish teeth and their digestive processes create the fine, white sand that characterizes many tropical beaches. This sand is chemically and physically different from sand produced by wave erosion, often being finer, whiter, and more uniform in grain size. Different parrotfish species contribute different grades of sand to beach systems, creating the diverse textures found on tropical shores. Smaller species like the striped parrotfish produce finer sand, with grain sizes averaging 0.17 millimeters, while large species, like the humphead parrotfish, create coarser particles up to 1 millimeter in diameter. The mix of particle sizes from different parrotfish species helps create the varied textures of tropical beaches, from powdery soft sand to coarser, more stable substrates. This variation in sand production affects beach dynamics, with finer sands being more easily transported by currents, while coarser sands create more stable beach formations. The color of sand can also vary depending on which coral species are being consumed, with some parrotfish preferentially feeding on specific corals that produce distinctively colored sand. This sand production is essential for maintaining beach and island systems in coral reef areas where terrestrial sources of sand are limited. Without constant replenishment from parrotfish and other bio-eroding organisms, tropical beaches would disappear over time as existing sand is transported away by currents and waves. Studies have shown that parrotfish can contribute up to 85% of the sand supply to some tropical beaches, making them the primary architects of these coastal formations. On some islands, virtually all the sand is of biological origin, with parrotfish being the dominant producers. The rate of sand production by parrotfish can exceed the rate of erosion, allowing beaches and even entire islands to grow over time. The relationship between parrotfish, coral, and beaches demonstrates the interconnectedness of reef ecosystems in ways that extend far beyond the underwater realm. Protecting parrotfish populations is essential not only for reef health, but also for maintaining the beaches and islands that support tourism and protect coastal communities. The sounds of parrotfish feeding a constant crunching and grinding that forms part of the acoustic signature of healthy coral reefs is literally the sound of islands being built. When parrotfish populations decline due to overfishing, beach erosion accelerates, coastal properties become vulnerable to storms, and the economic foundation of many tropical nations is threatened. Parrotfish have evolved specialized digestive systems to process large quantities of calcium carbonate while extracting maximum nutrition. Their pharyngeal mill, a set of grinding teeth in their throat, pulverizes coral fragments into particles fine enough to extract embedded organic matter. The efficiency of this system allows parrotfish to extract nutrients from coral while processing enormous amounts of limestone, up to 5 tons per year for large individuals. The digestive process involves multiple stages of grinding and acid treatment that break down coral skeleton while preserving organic compounds. This specialization represents millions of years of evolution, creating one of nature's most effective recycling systems. The feeding behavior of parrotfish also helps maintain coral health by removing algae that could otherwise smother coral colonies. This grazing pressure is essential for preventing algae overgrowth, particularly in areas where nutrients from human activities promote algae growth. Parrotfish feed selectively, targeting areas with the highest algae biomass while avoiding healthy coral tissue in most cases. Their feeding creates a mosaic of grazed and ungrazed areas that promotes biodiversity by preventing any single species from dominating. The constant grazing maintains a balance between coral growth and algae colonization that is essential for reef health. Different parrotfish species have different feeding preferences and behaviors, creating diverse impacts on coral communities and sand production. Some species focus on specific types of algae, using their keen eyesight to identify preferred food sources, while others are more generalist feeders that consume a wide variety of organisms. Excavating species actively bite into coral skeleton to access endolithic algae, producing more sand, while scraping species remove only surface material, producing less sand but causing less damage to coral colonies. The diversity of feeding strategies among parrotfish species ensures that sand production continues even if some species decline. The role of parrotfish in beach formation extends beyond sand production to include the creation of different beach morphologies and habitats. The size and distribution of sand particles produced by different parrotfish species influence beach slope, stability, and erosion patterns. Fine sands create gentler beach slopes that support different organisms than coarse sand beaches. The sorting of sand particles by wave action creates distinct zones on beaches, each supporting different communities of organisms, from ghost crabs to nesting sea turtles. Recent research has revealed that coral reefs may be able to hear approaching weather systems through changes in underwater sound patterns, demonstrating sensory capabilities we're only beginning to understand. The acoustic environment of coral reefs is surprisingly complex, filled with sounds from fish feeding, crustaceans snapping, and waves breaking on the reef structure, a symphony of biological and physical processes. This underwater soundscape carries information about environmental conditions, predator presence, food availability, and weather patterns that reef organisms have evolved to interpret. The discovery that reefs and their inhabitants may use sound to detect approaching storms adds another dimension to our understanding of how these ecosystems function and survive extreme weather events that could otherwise devastate unprepared communities. Changes in wave patterns and water movement associated with approaching storms create distinctive acoustic signatures that propagate through reef systems hours or days before the storms arrive. Low-frequency sounds produced by distant storm waves can travel hundreds of kilometers underwater, arriving at reefs long before any visible signs of approaching weather. Some researchers believe that reef organisms may use these sound cues to prepare for severe weather events by seeking shelter, reducing feeding activity, or altering reproductive behavior. The ability to detect storms in advance could mean the difference between survival and destruction for many reef species. Fish and mobile invertebrates that can sense these acoustic warnings have time to find secure shelter, while sessile organisms like corals may retract their polyps and increase mucus production for protection. Fish behavior on coral reefs often changes dramatically in the days before major storms, with many species seeking shelter in deeper reef crevices or moving to more protected areas with remarkable consistency. While some of this behavior may be responses to changes in barometric pressure that can penetrate water to significant depths or water temperature fluctuations, acoustic cues may also play a crucial role in these preparations. Studies have documented fish aggregating in caves and under overhangs up to 72 hours before hurricane arrival, suggesting they detect storm approach well before human weather instruments at that location. The coordination of these movements across entire reef communities implies a shared sensory system that allows different species to respond to the same environmental cues. The sounds produced by healthy coral reefs also serve as navigation beacons for marine life across vast ocean distances. Fish larvae settling on reefs appear to use reef sounds to locate suitable habitat from considerable distances, swimming toward the biological symphony of snapping shrimp, feeding fish, and other reef inhabitants. The characteristic sound signature of healthy reefs, a constant crackling, popping, and grinding, can be detected by some fish species from over 10 kilometers away. This acoustic beacon guides not just fish but also invertebrate larvae, sea turtle hatchlings, and even some marine mammals to appropriate habitats. The frequency composition and temporal patterns of reef sounds provide information about reef health, species composition, and habitat quality that influences settlement decisions. As coral reefs decline, their acoustic signatures change, potentially disrupting the sound-based navigation and communication systems that many reef species depend on for survival and reproduction. Degraded reefs produce less sound, up to 70% reduction in some frequency ranges, and different frequency patterns than healthy reefs. The loss of sound-producing organisms, like snapping shrimp and parrotfish, and the structural complexity that creates reef acoustics through echo and resonance, may have far-reaching consequences for reef ecosystem function. Quieter reefs attract fewer larval settlers, potentially creating feedback loops where degradation leads to reduced recruitment, which further degrades the reef. The study of reef acoustics has revealed that different reef habitats produce distinctive sound signatures that vary predictably with environmental conditions. Healthy coral areas generate different acoustic patterns than degraded areas, sandy bottoms, or seagrass beds, with peak frequencies, sound intensity, and temporal patterns, all varying between habitat types. This acoustic diversity provides important information that marine organisms use for habitat selection, navigation, and behavioral timing. The dawn and dusk choruses of reef fish create temporal landmarks that organisms use to coordinate activities, while the constant background of invertebrate sounds provides spatial information about reef structure. Research has shown that the acoustic environment of coral reefs changes dramatically between day and night, with different suites of organisms producing sounds during different periods. Daytime is dominated by fish feeding sounds and sea urchin grazing, while nighttime features more snapping shrimp activity and the sounds of nocturnal predators. This temporal variation in reef acoustics provides additional information that organisms can use for timing behavioral activities, from feeding to reproduction. The predictability of these acoustic patterns allows reef organisms to anticipate daily and seasonal changes, optimizing their activities for maximum success. The mechanisms by which reef organisms detect and respond to acoustic cues are still being studied, but appear to involve multiple sensory systems. Some fish species have specialized hearing organs, including otoliths earstones that vibrate in response to sound that are particularly sensitive to the frequency ranges produced by reef sounds. Invertebrates may detect sounds through mechanoreceptors that sense water particle movement, while some organisms may feel sounds through lateral line systems that detect water pressure changes. The diversity of sound detection mechanisms ensures that acoustic information is available to most reef organisms, regardless of their evolutionary history. Understanding reef acoustics has practical applications for reef monitoring and conservation that are already being implemented. Acoustic monitoring can provide continuous, non-invasive information about reef health and biodiversity that complements traditional visual survey methods. Underwater recording devices can operate continuously for months, capturing rare events and documenting changes in reef communities that would be missed by periodic visual surveys. The acoustic signatures of different species allow researchers to track population changes, spawning events, and responses to environmental stressors without disturbing reef organisms. While sexual reproduction and larval dispersal are important for coral populations, many coral species also reproduce asexually through various forms of cloning that can theoretically continue forever, achieving a form of biological immortality. This ability to self-replicate allows successful coral genotypes to dominate suitable habitats and persist through environmental challenges that might eliminate sexually reproducing populations. Clonal reproduction represents one of evolution's most successful strategies, allowing organisms to multiply rapidly when conditions are favorable while maintaining genetic combinations that have proven successful. In coral reefs, this strategy has created vast networks of genetically identical colonies that may be thousands of years old, having persisted through ice ages, sea level changes, and countless storms through their ability to regenerate and reproduce without sex. Fragmentation is one of the most common forms of coral cloning, turning what might seem like disaster into opportunity. When storms, predators, boat anchors, or other disturbances break pieces off coral colonies, these fragments can often reattach to hard surfaces and grow into new colonies that are genetically identical to their parents. This process can be remarkably efficient. Studies have shown that up to 90% of fragments from some species can successfully establish new colonies under favorable conditions. Some coral species appear to rely primarily on this form of reproduction, rarely engaging in sexual spawning, suggesting that cloning provides sufficient genetic success for certain ecological strategies. The fragments can survive drifting in currents for weeks, settling in new locations far from the parent colony, effectively allowing immobile organisms to colonize new territories. Budding is another form of coral asexual reproduction, where new polyps, or even entire small colonies, develop from the tissues of parent colonies through a process similar to how plants produce runners. These buds may eventually detach and establish independent colonies, carried by currents to new locations, or they may remain connected to form ever-expanding clone networks that can cover hundreds of square meters. Some massive coral colonies that appear to be single individuals are actually networks of clones that have grown together over centuries, sharing resources and coordinating responses to environmental challenges. The budding process can be triggered by environmental stress, seasonal cycles, or damage to the parent colony, providing a flexible reproductive strategy that responds to conditions. Some coral species can also reproduce through polyp bailout, where individual polyps abandon a dying colony and swim away to establish new colonies elsewhere, a last resort survival strategy. This emergency reproduction strategy allows coral genotypes to survive colony-level catastrophes, such as severe bleaching, disease outbreaks, or burial by sediment, and potentially recolonize areas after disturbances. The bailed-out polyps can survive for days in the plankton, using stored energy reserves while searching for suitable settlement sites. Once settled, a single polyp can establish an entire new colony through asexual reproduction, potentially rebuilding a reef from a single surviving individual. The immortal potential of coral clones means that some coral genotypes may be thousands of years old, perhaps even older, persisting through countless environmental changes by continuously creating new copies of themselves across reef systems. This clonal reproduction strategy provides stability and resilience, allowing successful genetic combinations to persist through time, but may also reduce genetic diversity and limit adaptive potential in changing environments. The trade-off between genetic stability and adaptability is one of the fundamental tensions in evolution, and different coral species have evolved different balances between sexual and asexual reproduction. Clonal reproduction in corals can create vast networks of genetically identical colonies spread across large areas of reef, sometimes covering several hectares. These clone networks may function as superorganisms, potentially sharing resources and information through various mechanisms that scientists are still working to understand. Chemical signals may travel between clone members, warning of predators or disease, while nutrients might be shared during stressful periods when some clone members are struggling. The coordination between clone members can be remarkable. They often spawn simultaneously and respond to threats in unison, and may even share metabolic resources through fusion of tissues where colonies meet. The balance between sexual and asexual reproduction varies among coral species and environmental conditions, creating diverse reproductive strategies across reef systems. Some species reproduce primarily through cloning in stable environments where their existing genetic makeup is well suited, but switch to sexual reproduction when conditions become stressful or when genetic diversity becomes important for adaptation to changing conditions. This reproductive flexibility allows corals to optimize their strategy based on current and anticipated future conditions, maximizing both short-term survival and long-term evolutionary potential. The advantages of clonal reproduction include the ability to rapidly colonize suitable habitat without waiting for rare, successful sexual reproduction events, and the preservation of successful genetic combinations that have been tested by natural selection. However, the lack of genetic diversity in clonal populations may make them vulnerable to diseases that can spread rapidly through genetically identical individuals, and environmental changes that affect one clone member will likely affect all. This vulnerability has become increasingly important as climate change creates novel, environmental conditions that existing genotypes may not be adapted to handle. Understanding coral cloning has important implications for reef restoration and conservation, providing tools for rapid reef recovery. Clonal reproduction allows restoration programs to quickly establish new coral populations using fragments from healthy colonies, a technique called coral gardening that has become central to reef restoration efforts. However, restoration programs must balance the speed of clonal propagation with the need for genetic diversity, often combining clonal reproduction with sexual reproduction techniques to maintain adaptive potential. Environmental factors can dramatically influence the rate and success of clonal reproduction in corals. Temperature affects metabolic rates and energy availability for reproduction. Light levels influence the energy available from photosynthetic symbionts. Nutrients can limit or enhance growth rates, and physical disturbance can create fragments while also potentially destroying them. Understanding these factors is crucial for predicting coral population dynamics and designing effective conservation strategies that work with natural reproductive processes rather than against them. And so, we reach the end of our journey through the whispered wonders of coral reefs. Though in truth, there is no real ending to these stories that have been unfolding for hundreds of millions of years. From the microscopic polyps that build underwater cities to the vast networks that influence global weather patterns, we have discovered that coral reefs are far more than beautiful underwater landscapes. They are living libraries of cooperation, written in calcium carbonate, chemical laboratories of survival, where nature experiments with the boundaries of life, and symphonies of existence that have played for millions of years and will continue long after our own species' brief movement has ended. Each discovery we've explored tonight reveals layers of complexity that scientists are only beginning to understand, reminding us that nature's innovations far exceed human imagination. Each coral colony we have encountered carries within its calcium carbonate walls the accumulated wisdom of countless generations, a genetic and structural legacy that stretches back through geological time. Every fish that darts between the branches, following ancient pathways carved by millions of predecessors. Every cleaner shrimp that tends to its clients in rituals older than human civilization. Every spawning event that fills the water with tomorrow's possibilities, like snow made of hope. All are notes in an ancient song of interconnection and resilience. These behaviors and relationships have been refined over evolutionary timescales that dwarf human history, creating systems of such elegance and efficiency that our most advanced technologies pale in comparison. These underwater gardens teach us that beauty and function are inseparable, that cooperation often triumphs over competition in the long game of survival, and that the most complex systems can emerge from the simplest rules repeated with patience and persistence. In their crystalline depths, we find reflections of our own need for community, our own dependence on unseen partnerships with the microbes in our bodies and the ecosystems that sustain us, and our own capacity for both creation and destruction. The coral reef stands as nature's greatest example of emergence, the principle that simple interactions between simple components can create complexity and beauty beyond imagination. The coral reefs continue their patient work in the darkness, building tomorrow from the calcium dreams of today, hosting millions of small lives in their eternal embrace. Their stories whisper of resilience and beauty that persists through catastrophe, of cooperation and wonder that creates abundance from scarcity, of a world beneath the waves, where life creates its most magnificent cathedrals, not through conscious planning, but through the accumulated actions of countless individuals working toward their own simple goals. Each polyp seeks only to feed, grow, and reproduce, yet together they create structures that alter ocean currents, influence weather, and support biodiversity that rivals any ecosystem on Earth. Each polyp that secretes its limestone home, one molecule at a time, each fish that finds shelter in coral branches and returns the favor through its waste, each spawn that drifts on ocean currents, carrying genetic information across vast distances, all carry forward the ancient wisdom of reef communities. They remind us that survival is not just about individual strength or even species success, but about the intricate webs of relationship that bind communities together across scales from the molecular to the ecosystem level. The reef teaches us that competition and cooperation are not opposites, but complementary forces that together create resilient, diverse, and beautiful systems. In the gentle sway of sea fans that bend with currents rather than breaking, we see the dance of life responding to ocean forces with grace rather than resistance. In the chemical battles between competing corals, slow-motion wars, invisible to our eyes, we witness the eternal drama of territorial conquest and the constant negotiation for space and resources that shapes all ecosystems. In the synchronized spawning that transforms reef waters into snow globes of reproduction, we observe nature's most spectacular acts of faith in the future, as billions of tiny packets of genetic possibility are released into the vast uncertainty of the ocean. These reef cities teach us about time itself, how the accumulated actions of countless small lives can build monuments that endure across geological ages, outlasting mountains and witnessing the drift of continents. They show us that patience and persistence, working together over timescales we can barely comprehend, can create structures of breathtaking beauty and complexity. A single coral colony growing 1 centimeter per year seems insignificant, yet given centuries or millennia, it becomes a massive structure that provides homes for thousands of species and protects entire coastlines from destruction. The ancient corals that have grown for millennia speak to us of continuity across timescales that dwarf human experience, connecting us to deep time in ways that challenge our perception of permanence and change. A single coral colony may have begun growing when human civilizations were taking their first tentative steps toward agriculture, and it continues its patient construction today, adding new chambers to its limestone mansion with each passing day. These ancient beings have witnessed the rise and fall of empires, the evolution of new species, and changes in ocean chemistry and temperature that would have seemed impossible to predict. The partnerships we have discovered between corals and their zooxanthellae algae demonstrate that cooperation can create abundance from scarcity, turning nutrient-poor waters into thriving ecosystems. In these waters, where nitrogen and phosphorus are vanishingly rare, this ancient alliance transforms sunlight into sugar, supporting entire ecosystems through the miracle of photosynthesis. The green partners work within their coral homes, converting light and carbon dioxide into the energy that powers reef cities, while the coral provides protection, nutrients, and an optimal environment for photosynthesis. This partnership has persisted through mass extinctions and climate upheavals, demonstrating that cooperation can be more durable than competition. The cleaning stations scattered throughout reef systems reveal sophisticated service economies that would impress any urban planner or economist. Fish wait patiently in organized queues for the services of tiny cleaner wrasses and gobies, suppressing their predatory instincts to participate in these underwater health spas. The trust and cooperation required for these relationships, where predators allow small fish to enter their mouths and gills, demonstrate that reef communities are held together by bonds far more complex than simple predator-prey relationships. These cleaning stations operate according to unwritten rules that all participants understand and follow, creating order from what could be chaos. As you drift towards sleep, let your mind wander through coral gardens, bathed in filtered sunlight that creates cathedral light through the water, where fish move like living rainbows, painting the sea with their colors. An ancient wisdom grows grain by grain in structures that will outlast our civilizations. Imagine the gentle current carrying away the day's concerns, like sediment swept from the reef, the rhythmic breathing of the ocean matching your own slowing breath as you sink into rest. Feel yourself becoming part of this ancient rhythm, your consciousness dissolving into the vast blue that has cradled life since the beginning. Picture yourself floating above a reef at twilight, that magical hour when the day shift of fish gives way to the night shift in a changing of the guard that has occurred every evening for millions of years. The butterfly fish and tangs that browse the reef during daylight hours retreat to their nighttime shelters, while moray eels emerge from their caves. Octopuses unfold from impossibly small crevices, and sharks begin their nocturnal hunts. Watch as coral polyps begin to extend their feeding tentacles, transforming the reef from a garden of stone into a constellation of feeding arms that dance in the current. The water is warm and buoyant, supporting your weightless form as you drift among these underwater cathedrals that rise from the seafloor like dreams made solid. The sounds of the reef create a natural lullaby, the gentle breaking of waves on the reef crest, like distant applause, the melodic calls of fish that sound like whispers in an ancient language, the soft whisper of water flowing through coral branches like wind through leaves. These are the sounds that have accompanied life on Earth for hundreds of millions of years, an oceanic soundtrack that has witnessed the rise and fall of countless species, the movement of continents, and the evolution of consciousness itself. This symphony continues whether we listen or not, a reminder that the natural world exists independent of human observation. In the crystal clear water above the reef, you can see down into the blue depths where deep-sea corals grow in eternal darkness, proof that life's creativity knows no bounds. These cold water architects build their cities without sunlight, sustaining themselves on the marine snow that drifts down from the surface world above like manna from heaven. They remind us that life finds ways to thrive in the most unlikely places, that the patterns of growth and community we see in shallow reefs are fundamental organizing principles that transcend specific environments. The parrotfish work as underwater landscapers, their constant crunching and grinding, slowly transforming coral skeletons into the white sand that will one day form tropical beaches where children will play and lovers will walk. They are the architects of coastlines, creating new land from the bones of ancient reefs, participating in geological processes that operate on timescales beyond human comprehension. Every grain of sand on a tropical beach has a history, having passed through the digestive system of a parrotfish that extracted life from stone. Night brings its own wonders to the reef, a complete transformation that reveals the reef's dual nature. Bioluminescent plankton create trails of blue-green fire, when disturbed by swimming fish, writing temporary messages in light that fade like thoughts. Fluorescent corals emit their hidden light shows, glowing pink and green and orange under moonlight, revealing patterns and communications invisible to our daylight-adapted eyes. These lights speak of processes we don't yet understand, of communications between species that predate language, of beauty that exists independent of observation. The reef's recycling systems work tirelessly through the night, breaking down waste products and redistributing nutrients through the complex web of life with an efficiency that human cities can only dream of achieving. Nothing is wasted in these efficient ecosystems, where every death feeds new life. Every exhalation becomes another organism's breath, and even waste becomes resource. The sponges filter millions of gallons of seawater, removing bacteria and organic particles while maintaining the crystal clear conditions that allow sunlight to penetrate to photosynthetic organisms below, creating clarity from chaos through patient, continuous work. Rest now in the knowledge that life continues its patient work in the coral cities beneath the waves, building structures of impossible beauty through the simple accumulation of small acts. Let the ancient rhythms of the reef carry you into dreams as deep and colourful as the gardens themselves, where time moves differently and small actions accumulate into monuments. The eternal reef symphony continues, playing its songs of cooperation, survival, and wonder into the infinite ocean night, a lullaby older than memory that will continue long after we are gone. The coral spawning events that occur on certain nights transform the reef into an underwater snowstorm of reproduction, a blizzard of potential futures. Millions of coral polyps release their eggs and sperm simultaneously, creating feeding frenzies that affect every level of the marine food web, from the smallest plankton to the largest whale sharks. These spectacular events remind us that even the most ancient and established ecosystems depend on successful reproduction to maintain themselves across time, that continuity requires constant renewal, that life must always be betting on tomorrow, even when today seems secure. As we have learned, coral reefs influence weather patterns far beyond their underwater boundaries, reaching into the sky to shape clouds and storms. The shallow waters above reefs warm and cool differently than deeper ocean areas, creating temperature gradients that drive local wind systems and cloud formation that bring rain to distant lands. The Great Barrier Reef extends its influence into the atmosphere above, helping to shape the climate of an entire continent, reminding us that the boundaries we draw between ecosystems are human conveniences that nature ignores. The deep-sea coral communities, growing in eternal darkness, remind us that reef ecosystems are not limited to shallow tropical waters, but represent a fundamental pattern of life organization. In the crushing depths where no sunlight has ever penetrated, coral architects continue their patient work, building structures that rival their sunlit cousins in complexity and beauty. These hidden reefs challenge our assumptions about where life can thrive and what conditions are necessary for complex ecosystems to develop, expanding our understanding of life's possibilities. The fish that change sex in response to social conditions demonstrate the remarkable flexibility of reef life, the ability to adapt not just across generations, but within individual lifetimes. In the spatially constrained environment of coral territories, the ability to switch between male and female roles ensures that reproductive opportunities are not wasted and that populations can maintain themselves even when environmental changes disrupt established social structures. This flexibility challenges our assumptions about the fixed nature of biological systems. The coral mucus that coats reef surfaces serves as medicine, food, and protection for countless reef inhabitants, a substance so valuable that entire communities of organisms have evolved to exploit it. This often invisible substance contains antimicrobial compounds that fight diseases, nutrients that feed small organisms, and chemical signals that facilitate communication between species. The study of coral mucus has led to discoveries that may benefit human medicine, as well as our understanding of reef ecology, reminding us that nature's pharmacy contains remedies we have yet to discover. The remarkable longevity of some coral colonies connects us to deep time in ways that few other living organisms can achieve. To encounter a coral colony that began growing thousands of years ago is to touch a living link to the distant past, a being that has witnessed the rise and fall of human civilizations while continuing its patient work of building and growing. These ancient organisms remind us that our perception of time is limited, that processes operating on geological timescales shape the world in ways we can barely perceive. The mobile corals that can move across reef floors demonstrate that even seemingly sessile organisms may be more dynamic than we realize, actively choosing their positions rather than passively accepting where they settle. These wandering architects search for optimal conditions, escaping from unfavorable situations and positioning themselves for maximum growth and survival. Their movements, though slow by human standards, represent active choices that can determine their fate over their long lifespans, teaching us that mobility comes in many forms. The chemical warfare between competing coral colonies reveals that reef communities are not as peaceful as they appear, that beneath the serene beauty lies constant competition for space and resources. The slow-motion battles for territory involve sophisticated weapons and defensive systems that have evolved over millions of years. These invisible conflicts shape the structure and composition of entire reef systems, determining which species dominate prime real estate and which must find refuge in marginal areas, creating the complex mosaics of species that make reefs so diverse. The acoustic world of coral reefs is filled with sounds that carry information about ecosystem health, approaching weather systems, and the availability of suitable habitat for new generations. The biological symphony produced by healthy reefs serves as a beacon that guides fish larvae and other marine organisms to suitable settlement sites across vast ocean distances. This hidden dimension of reef communication reminds us that there are entire sensory worlds that exist beyond human perception, that nature operates through channels we are only beginning to discover. The clonal reproduction strategies of many coral species provide a form of biological immortality that allows successful genotypes to persist across geological timescales. Through fragmentation, budding, and other asexual processes, corals can theoretically maintain their genetic lineages forever, creating vast networks of identical colonies that function as superorganisms spanning entire reef systems. This ability to clone themselves indefinitely challenges our concepts of individuality and death, showing us that life has found many solutions to the challenge of persistence through time.